Hello, Doc.
Yeah, hello, good morning.
Good morning,
Welcome
sir.
to
Good morning, sir.
this morning's lecture. Sorry for the hitch-up, but definitely get the ball rolling. This morning we'll be looking at DNA replication, its mutation, and repair. DNA replication, mutation, as well as the repairing of DNA. So without wasting much time, let's move on to the second objectives. You should be able to explain semi-conservative nature of DNA replication as the first objective. Then again, you should be able to explain DNA replication in eukaryotes and prokaryotes. Now, to begin with, semi-conservative DNA replication. DNA replication is an catalytic function of DNA, and it usually occurs during the S phase. You remember the phases we went through last week, SM, D, and the rest. Usually occurs during the S phase of cell cycle when chromosomes are highly extended, in highly extended form. That is when you drag it, that is when you'll be able to see it. So as proposed by Watson and Crick in their DNA replication model. So DNA replication is semi-conservative. It's said to be semi-conservative because each strand, the DNA double helix structure, serves as a template for the synthesis of a new complementary DNA strand. We talked about last week on zipping. I made emphasis to the fact that you pull up a zip, that means you are closing it. When you open it up, that means it is replicating. So it serves as a template for the synthesis of a new complementary DNA strand, and hence the newly replicated chromosome consists of one old and one new DNA strand. So as it unwinds, one stays in the nucleus, whilst the other, which leaves as a messenger RNA, moves into the cytoplasm to pair with other ribosomes to begin protein synthesis. So in the semi-conservative replication, the two strands would separate from one another, maintain their integrity, and each one will synthesize. And each one will synthesize. And each one will synthesize from the pool of nucleotides a complementary strand. I'll take that again. In the semi-conservative replication, the two strands would separate from one another. They maintain their integrity because of the codons that they bear, and each one will synthesize from the pool of nucleotides its complementary strand. So the result will be that the newly synthesized molecule would carry or conserve one of the two strands from the parent molecule, and the other strand will be newly assembled as depicted in the diagram that I'm about to show to you, figure 1.1. So there is sufficient evidence to prove that double-stranded DNA really replicates by a semi-conservative method. And here we go. You realize the replication, the double helix structure then begins to unwind. The portion in the black and the new strand. Okay, so the old strand, the original strand is in the ash coated. We realize that it begins opening up, unzipping with the codons, nitrogenous bases being exposed. Then comes in the new one, parental DNA showing the semi-conservative pattern of replication. Replication simply means that it's about to multiply. Okay, unzips and replicate. That means they are producing mirror copies of the DNA replication. So Okazaki is an ancient Japanese, suggested that DNA synthesis proceeds simultaneously on both the DNA strands, utilizing the same enzyme that it uses in breaking up DNA polymerase in the form of small isolated fragments. So these segments are known as Okazaki pieces, and it consists of about 1,000 to 2,000 nucleotides. So these are joined together by the enzyme polynucleotide ligase, completing the formation of polynucleotide chain. So it breaks up, unwinds. The old strand unwinds, one leaves into the cytoplasm. So the one that is left, the old strand that is left, the new fragments come to join it, and that forms a polynucleotide. So here we go. Because DNA is always synthesized in the 5' and the 3' direction. So one of the strands will be discontinuous. Because one will be 5, 5 to 3, then 3 to 5 in that direction. So this is a lagging strand. Okay, according to this diagram. Just as we looked at other diagrams last week, the phosphate molecule, 3 phosphate and 5 phosphate. So DNA is always synthesized in a 5 to 3 direction, 5 to 3. That is how synthesis occurs with regard to DNA. So 5 to 3 has to be what? Discontinuous. That's in the opposite direction. Barely happens. It always has to do with 5' to 3'. This is referred to as what? A lagging strand. So J. Cairns, from his experiment, concluded that DNA synthesis starts at a fixed point on the chromosomes and proceeds in one direction. It starts at a fixed point, and the fixed point is from what? A 5' to 3'. Fixed point to in one direction to another point. So recent experiments suggest of a bi-directional replication. They started it earlier, they researched into it earlier. But modern day research is depicting that there is a bi-directional replication. While some is moving to the right, others are also moving the opposite direction. So Leventhal and Cairns proposed that during replication, the two strands do not separate completely before replication. The two strands, they do not replicate, separate completely, sorry, before replication. Instead, they start unzipping at one end, and simultaneously, the unzipped segments start attracting their nucleotide pairs. So the adenine will definitely attract the thymine, whilst the guanine will definitely attract the cytosine. So instead, they start unzipping at one end, and simultaneously the unzipped segments start attracting their nucleotide pairs. So in this way, the unzipping of the original DNA strands and the synthesis of the fresh strands go side by side. So as it is unzipping and exposing the nitrogenous bases, its complementary base pairings will definitely come and form a new or fresh DNA strand. Now let's look at DNA polymerase, that wonderful enzyme. It is the chief enzyme of DNA replication, DNA polymerase. It is the chief enzyme. Without DNA polymerase, replication wouldn't occur. It is actually what propels it to replicate. So its activity was first demonstrated by Kornberg in 1956, and it catalyzes the covalent addition of deoxyribonucleotides to the 3' phosphate hydroxy of a pre-existing nucleotide called a primer. So the DNA polymerase 1 enzyme is now considered to be a DNA repair enzyme. It's like you getting hit and the wound healing. After some time, you realize that, everything is sealed. So there is a repair. DNA polymerase 1 is now considered to be a DNA repair enzyme rather than a replication enzyme. So this enzyme is known to have 5 active sites, namely template site, primer site, 5' to 3' phosphate site, cleavage or exonucleate site, nucleoside triphosphate site, and 3 to 5' cleavage site, or 3 to 5 exonucleate site. So these are the 5 active sites that the DNA polymerase actually works on. Now, the replication apparatus for DNA polymerase 1 is mainly involved in removing ribonucleotide acid, RNA primers from Okazaki or the precursor fragments and filling the resultant gaps due to the 5' to 3' polymerizing capacity. So DNA polymerase 1 enzyme can also remove thiamine in RNA. Instead of thiamine, the removal, because DNA polymerase enzyme has acted on it, it is replaced with what? Uracil. So instead of the 4 complementary bases, you have adenine, guanine, cytosine, and thiamine. In RNA, we have adenine, guanine, cytosine, and uracil. Uracil replaces thiamine. So DNA polymerase 1 enzyme can also remove thiamine and primers, which are produced due to the UV radiation, and fill the gap due to excision. This is called proofreading or editing function of this enzyme. So instead of adenine pairing with thiamine in uracil, in RNA, it is the thiamine is replaced by uracil. DNA polymerase 2. This enzyme resembles DNA polymerase 1 in its activity.
Hello, sir.
But yes.
So if you can go back to the previous slide, I want to show you something. Yes, so the DNA polymerase 1 enzyme also removed thiamine. The word after thiamine, what word is that? Is it primerase or dimase?
Supposed to be primase.
And then you have ultraviolet radiation. It is irradiation.
Irradiation. Yes.
All right. Thank you, sir.
Welcome. So this enzyme resembles DNA polymerase 1 in its activity, but in a DNA repair enzyme, and it brings about growth in the 5 to 3 prime direction, 5 to 3 prime, using free 3 hydroxy groups, 3 phosphate hydroxy groups. DNA polymerase 3. We have done DNA polymerase 1, 2, and we are now on the third. It plays an essential role in DNA replication, and its multimeric enzyme or holoenzyme have 10 subunits. Has 10 subunits. And all these 10 subunits are needed for DNA replication in vitro. That means in person. However, all have what different functions. Each of them have what different functions. So for example, alpha unit has what, let's say 3 prime to 5 prime exonuclease, proofreading or editing activity. Then the core enzyme comprises 3 subunits, alpha, beta, gamma. These are core. But in reality, it has what, 10 subunits. So the remaining 7 subunits increase possessivity. That means rapidity, efficiency with DNA polymerase, extending what? The chain. Each and every subunit has a major role that it plays with regards to the replication of DNA. Eukaryotic DNA polymerase. So eukaryotes, example, yeast, rat liver, human tumor cells are found to contain some 5 types of DNA polymerases. So we have alpha, DNA polymerase alpha, DNA polymerase beta. So with alpha, relatively, it has a high molecular weight enzyme, and it's also called cytoplasmic polymerase or large polymerase. It is found both in the nucleus and in the cytoplasm. Found both in the nucleus and the cytoplasm is as a result of the replication. When it first replicates, replication begins in the nucleus. Then the mirror copy leaves the nucleus into the cytoplasm. So that is why you are able to find DNA polymerase alpha in the cytoplasm as well as in the nucleus. DNA polymerase beta. It is also called nuclear polymerase or small polymerase, and is found only in invertebrates like the insects, the slugs, and other organisms. Only in invertebrates. DNA polymerase beta. DNA polymerase Y. This enzyme called mitochondrial polymerase, and it is encoded in the nucleus. Then DNA polymerase E. This enzyme is found in mammalian cells and is referred to as proliferating cell nuclear antigen, PCNA, dependent for DNA synthesis processivity. And the fourth, I think we are on the fourth, right? The fifth, sorry, with eukaryotic polymerases. It was previously known as DNA polymerase 5-2. And this enzyme is pCNA independent and occurs in mammalian HeLa cells and budding yeast. The large DNA polymerase A or alpha is the predominant DNA polymerase enzyme in eukaryotic cells and was believed for a long time to be the only enzyme involved in DNA replication. That is why we keep researching. We keep researching. Wherever you end your research, somebody carries it on because you cannot find everything. But now, earlier it was believed, this enzyme was believed to be the predominant DNA in eukaryotic cells for a very long time. But now, one more polymerase, namely DNA polymerase 8, is also found to be involved in eukaryotic DNA replication. So these DNA primers, these enzymes catalyze the synthesis of DNA primers, which are a prerequisite for the initiation of DNA replication in the vast majority of organisms. So before actual DNA replication starts, short RNA oligonucleotide segments called RNA primers, or simply the primers, have to be synthesized by DNA primase enzyme utilizing ribonucleoside triphosphates. And these RNA primers are synthesized by copying a particular base sequence from one DNA strand and differs from a typical RNA molecule in that after the synthesis, the primer remains hydrogen bonded to the DNA template instead of the polymerase, which actually divides it. So earlier we made mention of the fact that hydrogen bonds holds the DNA molecules together, the double-stranded DNA. Hydrogen bonds actually hold them together. So the primers are about 10 nucleotides long in eukaryotic cells, and they are made at intervals on the lagging strand, where they are elongated by DNA polymerase enzyme to begin each Okazaki fragment. So the Okazaki fragment, these are like blocks on the polynucleotide. Immediately it gets to a particular codon, there is a break, it continues. So that forms the Okazaki fragments. So these RNA are later excised and filled with DNA with the help of DNA repair system in eukaryotes. In bacteria, two different enzymes are known to synthesize the primer RNA oligonucleotide and RNA polymerase on the leading strand, and DNA primase on the lagging strand. So there is a leading strand which actually leads the strand. Okay, let's say the Okazaki fragment, the strand, and then a DNA primase that forms the base. That forms the base, the mirror copy. Then there's a leading strand, and there's a lagging strand, one that remains in the nucleus and the one that leaves into the cytoplasm. So polynucleotide ligase, this enzyme is an important enzyme both in DNA replication and in DNA repair. DNA ligase catalyzes the formation of phosphodiester linkage between the 5' phosphoryl group of the nucleotide and the 3-hydroxy group of the immediate neighbor at the side of a nick in a DNA strand. So it seals the nicks in a DNA strand. It seals the nick in the DNA strand. Earlier, I made you aware of the fact that there are 4 main nitrogenous bases: adenine, thymine, guanine, and cytosine. Adenine always pairs with what? Thymine. And guanine always pairs with cytosine. That is on the DNA molecule. But when it comes to the RNA molecule, the thymine is replaced by what? The uracil in that regard. Endonucleases, particularly restriction endonucleases, they restrict, are also important during DNA replication as well as DNA repair. So during replication, an endonuclease may produce a nick in the origin to initiate replication, or it may induce nicks to generate a swivel for facilitating DNA unwinding. Nicks are like triggers. You have a gun. If you don't pull the trigger, the bullet wouldn't come out. It is just lying there. Immediately you pull the trigger, that is when the bullet comes out, and you hear the rushing sound. Boom. So during DNA replication, endonucleases may produce a nick in the origin to initiate replication, or it may induce nicks to generate a swivel for facilitating DNA unwinding. Enzymes involved in opening of DNA helix. Enzymes involved in opening of DNA helix. So DNA helicases, they are ATP dependent unwinding enzymes, which promote separation of the 2 parental strands and establish replication forks that will progressively move away from the origin. So by that, unwinding of the DNA helix at the replication fork could in principle be catalyzed by 2 DNA helicases acting in concert. One running along the leading strand and the other along the lagging strand. Helix, the stabilizing strand, also called single strand DNA binding proteins or single strand binding proteins. So behind SSBPS, so behind the replication fork, the single DNA strands are prevented from rewinding about one another or forming double-stranded hairpin loops in each single strand by the action of single strand binding proteins, SSBPS. So SSBPS bind to expose DNA strands without covering the bases, which therefore remain available for the templating process. Top isomerases or DNA gyrases. The action of a helicase introduces a positive supercoil into the duplex, that's the DNA double helix structure. So it introduces a positive supercoil into the duplex DNA ahead of the replication fork. Enzymes called top isomerases. Isomers, they are just like a mirror copy of an original enzyme. They relax the supercoil by attaching the transiently supercoiled duplex, nicking one of the strands and rotating it through the unbroken strand. The nick is then resealed. One type of top isomerase that is the top isomerase 1, causes a single strand break or nick, which allows the two sections of DNA helix on either side of nick to rotate freely relative to each other, using the phosphodiester bond in the strand opposite the nick as a swivel point. A second type of top isomerase, that is top isomerase 2, forms a covalent bond to both strands of DNA helix at the same time, making transient double strand break in the helix. A replicon is the unit of DNA in which individual acts of replication takes place. It's just like a replica of an image. So a replicon is the unit of DNA in which individual acts of replication takes place. That is, it is capable of DNA replication independent of other segments of the DNA. So therefore, each replicon has an origin. There is an original in which replication is initiated called ori, as in origin. Okay, so we'll be looking at a figure very soon representing a replicon, and it may have a terminus somewhere that it ends its terminus, terminating, at which the replication stops. So this is the origin of DNA replication on mammalian chromosomes, and so origins of DNA replication. So every 150 kilobits. So like we know 5 prime starting and ending with what? 3 prime, 5, in that order. Okay, bidirectional. So you realize that at a point it begins with 5 and moves to 3, then the opposite occurs. That is why we have what? Bireplication. Initially, we were thinking it was unidirectional, but modern day research made us aware, as stated earlier in the slides, made us aware that replication is what? Bidirectional. So fusion of bubbles, replication of bubble. Initially, you realize that there is a small bump emerging. Then it bulges. Into 2 bubbles. Then you have what? Daughter chromosome. So they divide and form what? Daughter chromosomes. So instead of 2, you realize that we have what? 4, like what we did earlier with regards to cell division. In meiosis 1 and meiosis 2, at the end of the day, we have what? 4 daughter cells instead of 2 that occurs in what? Mitosis. So the bacterial and viral chromosomes usually contain a single replicon or chromosome because they divide very fast. Bacteria and viruses, they divide very fast. That is why corona was able to spread across the world in no time. The bacterial and viral chromosomes usually contain a single replicon or chromosome. So although stage 2 has 2 origins, 2 ori, 1 primary and 1 secondary, but in the presence of primary origin, the secondary origin is, as a rule, non-functional. The second is always non-functional according to the phage 2 process. So in E. coli, Escherichia coli, the point of origin is identified as the genetic locus, ori C, or original or origin C. In E. coli, the point of origin, the primary origin, is referred to or considered as what? The genetic locus. And
hello, sir.
To refer to, yes.
Hello, sir.
Hello.
Sir, please, sometimes can you be adding some sketches to explain for us?
What you have is what I give, please. Okay, so at certain points, that is when I bring in the diagrams.
But I
Is that
don't know
clear?
if I don't know if you can do some other sketches to maybe drawing it as we see with explanations.
Very well. Very noted. I've noted that. So the E. coli point is referred to as the genetic locus or origin C. A complete prokaryotic origin supports the following three functions. Initiation of the replication. Before anything begins, you need to plan. So there is an initiation process, initiation of the replication, and control of the frequency of initiation events. Whatever that you are about doing, you have control over it. So the same occurs in DNA replication. Like you writing your project work, your long essay or your thesis. Right? You first present a proposal, and if everything goes on well, you build on the proposal. It becomes your research work. So initiation of replication, you have control of the frequency of initiation events, and segregation, separate them. Segregation of the replicated chromosomes into the daughter cells. Origins have been identified in bacteria, in yeast, in chloroplast, and mitochondria. And their significant general feature is that they are AT rich, adenine thymine rich. Bacteria, yeast, chloroplast, and mitochondria, they are AT rich, adenine thymine rich, which may be important for easing unwinding during replication. So the bacterial origin contains many different short sites that are needed for its function. These sites are sometimes separated by specific distances, but not by specific sequences. So these specifically located sites are needed for the binding of different proteins involved in DNA replication. Several prokaryotic replicants have specific sites called terminals. So it has an initiation site as well as what? An origin, then a terminating site where it begins and where it ends. So several prokaryotic replicants have a specific site called terminals, which stops replication fork movement and thereby terminates DNA replication. So it begins at a certain point and ends. It has to be there for some time before it gathers enough materials and continues the process, the interface, just like the phases of mitosis and meiosis that we have. Each terminus is specific for one direction of fork movement. Each terminus is specific for one direction of fork movement. The T1 and T2 are arranged in such a way that each fork should have to pass the other to reach the terminus specific it. So the replication terminus requires the product of a gene, which probably codes for a protein that recognizes T and T2. In eukaryotes, each chromosome has several replicants. Example, yeast has 500, Drosophila has 3,500, mouse or the mice that are hovering around, they have 2,500, and Bifidobacter has 35,000. So in eukaryotes, cells with true nucleus, each chromosome has what? Several replicants, and the examples are as displayed. Now, at any given point during the S phase, only some of these replicants undergo replication. Each replicant seems to be activated at a specific time in a specific sequence. So the length of a eukaryotic replicon may vary from 40 kilobits in yeast and Drosophila to about 300 kilobits in Vishia. So the number of detectable replicons seems to vary with the developmental stage and the cell or tissue type. This is explained on the basis of tissue-specific origins, so that some origins are active in some tissues, while others are active in some other tissues. So this is exemplified by Drosophila, where the early embryonic cells have 10 times as many replicons as those in the adult somatic cells. Fidelity of replication. The error of DNA replication is much lower than that of transcription because of the need to preserve the meaning of the genetic message from one generation to the other. In a particular family, if you look at the genealogy, you realize that there's a particular trait running through all members of the family. The fact of it is it's being owed to fidelity of what? Replication. Error rate of DNA replication is much lower than that of transcription because of the need to preserve the meaning of genetic message from one generation to the other. So for example, the spontaneous mutation rate in E. coli is about 1 error per 1010 bases. So the error is very minimal, incorporated during replication. This is due primarily to the presence of the minor automeric forms of the bases which have altered base-bearing properties. That was why in the olden days, for instance, when genetics had not been so acclimatized with our ancestors, before they go in for marriages, they go finding out certain information about the family they're about to enter. Whether there are some diseases in that family, whether there are some problems inherent, that they have some untoward or ill-mannered character. They look at all these forms, and it's as a result of what? DNA replication. So the error rate is minimized by a variety of mechanisms. DNA polymerases will only incorporate an incoming nucleotide if it forms the correct base pair, the template nucleoside in its active site. So the occasional error is detected at the 3 to 5 prime through reading exonucleates, which is associated with the polymerase. If there is an error, it behaves on this particular hydroxy molecule and the 3 to 5 prime exonuclease to detect it. This removes the incorrect nucleotide from the 3 hydroxy end, 3 prime end, before any further incorporation. Immediately it detects an error. It behoves on this Yoko boss to scrutinize the error that is being detected. So this removes the incorrect nucleotide from the 3 prime end before any further incorporation, allowing the polymerase Then we insert the correct base. In order for the proofreading, sorry for the hold on. In order for the proofreading exonuclease to work properly, it must be able to distinguish a correct base pair from an incorrect one. The increased mobility of unanchored base pairs at the very 5' end of newly initiated lagging strand fragments of DNA means that they can never appear correct and so cannot be proofread. Hence, the first few nucleotides are ribonucleotides, RNA, so that they subsequently can be identified as low fidelity material and replaced with DNA elongated and proofread from the adjacent fragment. So errors which escape proofreading are corrected by a mismatch repair mechanism. There's a scrutiny occurring in the replication. So it tries as much as possible to minimize errors. Like, for instance, in modern day, let's say examination, like what Y does, we have script checkers, you have assistant examiners, you have team leaders, you have zonal leaders, you have chief examiners. The scripts go through all these phases before results are what keyed in to minimize errors. Hence, the first few nucleotides are ribonucleotides, that's RNA, so that they subsequently can be identified as low fidelity material and replaced with DNA elongated from the adjacent fragments, so that errors which escape proofreading are corrected by a mismatch repair mechanism. So the mechanism of DNA replication in prokaryotes in vitro means it goes on, and DNA replication has been extensively studied in E. coli. The mechanism of DNA replication in prokaryotes have been extensively studied in the bacteria Escherichia coli and in the phages and plasmids of E. coli. So in E. coli, the process of DNA replication involves the following three main steps. It comprises the recognition of the origin, ORI, the recognition of the origin, the primary source, then opening of the DNA duplex. That is when it starts unwinding to generate a region of what single-stranded DNA. Initiation of the DNA replication. You first target, or it first has to do with the origin, the primary source. Then the unwinding, opening of DNA duplex to generate a region of what single-stranded DNA, and capture of DNA B protein. DNA B protein. Capture of DNA B protein. That is 5' 3' helicase also acts as the activator of the primase. Also acting as the activator of the primase. That's the DNA initiator, DNA A or initiator protein. That is adenosine triphosphate complex binds at 9 bits. 9 bits. Inverted repeat regions. So it has certain fragments. So when it gets there, it inverts. There is an inversion. It turns upside down. Okay, and it repeats. So ABP complex binds at 9 BP inverted repeat regions. That is R prime RVR4 of the origin C, of the origin, the primary source. It promotes opening of the DNA duplex in a region of 3 direct repeats. That is, it started with 9, you have 13 BP sequence called 13 isomers or primers. The opening occurs from right 13 primers leftwards and requires negatively supercalled DNA and HU or IHF initiator proteins. All these are proteins, the HU and IHF. Then DNA B helicase. DNA B helicase. It is transferred to the exposed single-stranded DNA and causes unwinding of the DNA in the presence of ATP, SSB proteins, and DNA gyrase of isomers. All these are enzymes that are related to the DNA. So this results in unwinding. It is not only the DNA polymerase, but there are other enzymes which help the unwinding process. So this results in unwinding of the DNA duplex and the replication from origin C proceeds in both directions. Earlier, we made emphasis on the fact that early researchers were thinking of a unidirectional stage whereby it was occurring in one direction. But modern day scientists or researchers have made us aware that it occurs in both directions, which is bidirectional. So the replication from origin C proceeds in both directions. Then the SSB binding proteins occurs on a single-stranded region and the 2 DNA B complexes, that is primosomes, are loaded, one on each strand. The next figure gives an illustration which best explains the initiation process, how the whole process begins. So initiation of the DNA synthesis at the E. coli origin, ORI-C. So you have 5' then on the 3' and the 3' beginning in that order. Binding of what the DNA proteins, binding of the DNA proteins. So you realize that there's a ball emerging on the first stage. Then on the second stage, DNA proteins coalesce, they come together. They come together, coalescing. They come together. DNA melting induced by what? DNA proteins. So we have DNA B and DNA C proteins bind to single-stranded DNA. So they are coalescing. Then they further unwind. It further breaks up. Initially, they are together. Then there is a breakup. That informs the unwinding aspect. Elongation of the DNA molecule. So this step requires the presence of the following enzymes. Elongation in extending the DNA chain. So this step requires the presence of the following enzymes and factors. DNA B or helicase, also called mobile promoter. Then primase, DNA G. Then you have DNA polymerase, holoenzyme or DNA polymerase 3, HE, holoenzyme. Then RNase, ribonucleic acid, acidase, H, which removes RNA primase. Then DNA polymerase 1, which is used for filling the gap created due to RNA primase. And DNA ligase, which converts primer less Okazaki fragments into continuous strand. So during the initiation to elongation transition, the following events occur. Now let's look at the events occurring as a result of that. And with regards to the DNA polymerase, earlier in this lecture, I made you aware that there are 3 types of DNA polymerase: polymerase 1, polymerase 2, and polymerase 3. So as helicase or DNA travels in the 5 to 3 direction, it generates a replication fork by opening the DNA duplex. So the DNA strand having helicase becomes the lagging strand. It becomes a lagging strand. Then the DNA primase associates with DNA helicase, forming the primosome, which synthesizes multiple primers for lagging strand and single RNA primer for the leading strand. So there's always a lagging strand and a leading strand, the one which stays behind and the one which leads, because it is now beginning. So you have to move. So for the synthesis of lagging strand, the DNA polymerase 3 holoenzyme has to work on the same strand to which the DNA B helicase is bound, but it travels in opposite direction. That's DNA B helicase, DNA G primase, and DNA polymerase 3 work together in that strand elongation. Yes.
Please, what's the difference between the DNA B, which is in small letters, and the DNA that we know, which is in capital letters?
It is the same DNA, but to emphasize, to bring about the distinction, that is why they desire to put up in the small letters. Okay.
Okay.
So that it doesn't confuse you.
Oh, okay. So if I should write the capital,
because there are a lot of primers actually taking place in order to bring about the segregation, in order to bring about, sorry, the distinction. Okay, Okazaki decided to use the small letter E to represent the enzymes that he was working on. It is the same DNA, deoxyribonucleic acid, that you know.
Thank you, Doctor.
Is that all right?
Yes, please.
Very well. So helicase and DNA polymerase assembly remains processive. That is, they remain tightly bound to the replication fork and stay bound throughout the reaction. There goes the reaction. So you realize that, just as you were asking, the DNA G primase, okay, during the unwinding, right, DNA primase binds. DNA G, the primase, it binds. Then the DNA P further unwinds. One is binding and the other is what? Unwinding. It's in a process of what? Trying to elongate. So in order for it to elongate, it has to what? Unwind before it can go through the process of what? Elongation. So DNA P further unwinds, and the helix, it unwinds the helix, displaces DNA A proteins, right? The proteins are those are the ones that have been labeled AAA, and it synthesizes an RNA primer. Immediately it unwinds, the probability of an RNA forming is high because there's some which is going to stay in the nucleus. Then the others, the messenger, the copy of it move into the cytoplasm. That is when the synthesis of what? RNA begins. Okay, so it's no longer DNA, but it becomes what? mRNA, messenger. It is carrying a copy of the parent material into the cytoplasm, where it goes to, it starts its own elongation and, you know, binding process. So it synthesizes an RNA primer with regards to this. That's the elongation of DNA by an assembly of what? Proteins. An assembly of proteins. So the synthesis, that's elongation of lagging and leading strand, takes place by somewhat different methods. It is far more complex for lagging strand than for what? Leading strand. The one that stays behind, the lagging, it is lagging behind. And the leading strand normally informs the other enzymes, okay, to form messenger, ribosomal, and transfer RNAs. Discontinuous synthesis on lagging strand. The primase is taken up on solution and is activated by helicase, DNA, to synthesize an RNA primer that's 10 to 20 nucleotides long. 10 to 20 nucleotides long. So that is the action of the primase, on the lagging strand. Then the RNA primers are recognized by the DNA polymerase III holoenzyme on the lagging strand. And they are then utilized for synthesis of precursor or Okazaki fragments. So in fact, each new RNA primer is recognized by the gamma subunit of what DNA polymerase III holoenzyme and loaded with P units of the same polymerase. And this preloaded P subunits may then be captured or capture the core of the DNA polymerase III holoenzyme when it becomes available after finishing its synthetic job in the preceding Okazaki fragment. The long and short of it is that it begins the process. And you see, because of the interface stage of mitosis or meiosis, right? It gathers enough energy, enough enzymes to undergo the various processes. Prophase, metaphase, anaphase, and telophase. It gathers enough enzymes in that regard. So over here, it is the DNA polymerase III holoenzyme that it's actually utilizing. So on completion of the Okazaki fragments, the RNA primers are excised by DNA polymerase I. We realize that the previous one was talking about polymerase III, and now polymerase I, which then fills the resulting gaps with DNA. Then, after DNA polymerase I adds the final deoxyribonucleotides in the gap that is left by the excised primer. So the DNA ligase forms the phosphoester bonds. It forms the phosphodiester bond that links the 3' prime end of the primer replacement to the 5' end, the Okazaki fragment. So that has to do with discontinuous synthesis. Now we are moving on the continuous synthesis on leading strand. Continuous synthesis. Discontinuous was what we talked about. So in continuous, and this is in bidirectional DNA replication. The leading strand is primed once on each of the parental strands. On each of the parental strands. And 2, the RNA primer of the leading strand is synthesized by RNA polymerase enzyme. 3, DNA polymerase III holoenzyme causes elongation of the leading strand. And finally, DNA polymerase I and ligase enzymes give final touch to the leading strand, as in case of the lagging strand. As shown in the next, which is figure, is a DNA synthesis at the replication fork. So the direction, that's with regards to the arrows of leading strand synthesis and lagging strand synthesis, are in both the 5' to the 3' direction. So DNA polymerase moves continuously along the leading strand. So as the template DNA unwinds, exposing the single-stranded template for the lagging strand, the primerase has to synthesize an RNA primer, to which the DNA polymerase synthesizing the lagging strand can bind. As the DNA, as the replication fork moves further to the left, opening up more DNA, another RNA primer has to be synthesized. Each RNA primer dashed line on the lagging strand then serves as a starting point for the initiation of DNA synthesis. That's the Okazaki fragment that informs the solid line on figure 1.7 and 1.8. So the solid line occurring, Okazaki fragment. Okay, so the RNA primer is the one that is in broken lines, RNA primer, and the Okazaki fragment is the thick lines, that is indicated. So direction of leading strand synthesis, and we have the replication fork, just like the zipper example that I emphasized on, like a zip. So you have a zip, you pull up, you are closing, you pull down, you are opening. So since you are pulling down, because we are replicating, we are pulling down, we want to make more copies, you pull down, and you open up the nitrogenous bases for further coding, codons, further bonding. So replication movement of the fork, direction of movement of the fork. So you realize a 5' to 3' direction, it is forming RNA primase, Okazaki fragment, RNA primase, Okazaki fragment, and that is how it goes. So it further goes by showing this RNA primer, Okazaki fragment, DNA polymerase 3. So DNA polymerase 3 initiates as a primer. It is what begins the process of replication and elongates DNA up to the next RNA primer. Then newly synthesized DNA, Okazaki fragment occurring. So after the broken, you realize that it becomes what? Thickened. It becomes thickened. So polymerase 1, DNA polymerase 1 initiates at the end of Okazaki fragment. It further elongates the DNA chain while simultaneously removing the RNA primer with its 5' to 3' exonuclease activity. So elongation of DNA chain and removal of the RNA primer. So newly synthesized DNA as a Okazaki fragment, DNA ligase seals the gap by catalyzing the formation of the 3', 5' phosphodiester bond in an ATP-dependent reaction. So ATP is being formed here. Protein synthesis occurring as a result. So elongation of DNA chain as DNA ligase seals the gap to form the newly synthesized deoxyribonucleic acid. Now the replication fork. The replication fork is a structure that forms within the nucleus during DNA replication. It is created by helicases, which break the hydrogen bonds holding the two DNA strands together. The resulting structure has two branching prongs, each one made up of a single strand of DNA. These two strands serve as a template for the leading and lagging strands, which will be created as DNA polymerase matches complementary nucleotides to the templates. The templates may be properly referred to as the leading strand template and the lagging strand templates. So here we go. So the scheme of replication fork. So the template, you realize that A and the portion labeled A forms the template. Then the leading strand, the portion labeled what? B, 5 prime, okay, 5 prime going to 3 prime. The portion labeled B is the leading strand. Then the lagging strand C, the portion labeled what? C is the lagging strand. Then D informs what? The replication fork. D, replication what? Fork. It is what? It is doubling. Okay. You realize that all those ones are single, but immediately it got to D, replication, it is doubling and it's coming together. Then E is a primer. E is a primer. And the Okazaki fragment that has been formed informs the Okazaki fragment that has been formed. DNA replication in eukaryotes. So eukaryotic DNA replication requires two different DNA polymerases, namely DNA polymerase A and DNA polymerase gamma. So DNA polymerase gamma synthesizes the DNA on the leading strand, continuous DNA synthesis, whereas DNA polymerase A synthesizes the DNA on the lagging strand. One synthesizes DNA polymerase on the leading strand, and the other synthesizes DNA on the lagging strand. So there is an origin, just like the primary source and the terminus, one that ends, okay, beginning and end. But here, the beginner is the lagging. I have the mother. I have produced something and I'm lagging behind. Then you have to take the lead because you are new. I've been through everything. Since you are new, you have to take the initiative, move. You cannot stay on with me. So that informs the leading strand. So DNA replication in eukaryotes. These two enzymes and DNA replication actually helps. So you have T antigen and the replication factor E, also called eukaryotic SSB protein. Then the top isomerase 1, top isomerase 2, the proliferating cell nuclear antigen PCNE, also called cyclic, and the replication factor C or RFC. These are the stages or terms that one is supposed to have with regards to DNA replication in eukaryotes. So the steps we are taking one after the other. So before the onset of DNA synthesis, there is a pre-synthetic stage of 8 to 10 minutes duration for the formation of unwound DNA complex. And this step needs only 3 purified proteins, namely the T antigen or tag antigen or tumor antigen or RF, top isomerase 1 and 2. So this is how DNA replication in eukaryotes begins before the DNA synthesis. The tag antigen or the T antigen, using its DNA binding domain, forms a multi-subunit complex. And earlier we made emphasis on the fact that there are about 10 of those subunits with site 1 and site 2 in the presence of ATP and caused local unwinding. So more extensive duplex unwinding occurs due to association of the RFA and the top isomers with the help of DNA helicase component of the D antigen. The top isomers help in unwinding of the DNA by altering the topology of DNA, the replication form. So it helps to break the duplex by altering DNA. The RFA or SSB proteins bind to unwound single-stranded DNA. They bound to unwound single-stranded DNA. So after it has produced a mirror copy, there are certain antigens or antibodies that hovers around and they align themselves to the mother copy, and it's good to form the helix again. So the prime RNA synthesis is performed by primase, which is tightly associated with DNA polymerase X. DNA polymerase helps in synthesis of an Okazaki fragment in a 5-to-3 direction. Then replication factor RFC or PCNA cycling help in switching of the DNA polymerases so that polymerase A is replaced, polymerase A is replaced by polymerase 5, which then continuously synthesize DNA on the leading strand. Another Okazaki fragment is then synthesized in the replication fork on the lagging strand by a polymerase A. Primase complex, and this step is repeated again and again till the entire DNA molecule is covered. Then the RNA primers are removed and the gaps are filled as in prokaryotic DNA replication. Now, on the verge of the review, DNA replication occurs during the S phase.
Hello, Doc.
Yes.
Please, these replications and the DNA formation, is it that it occurs throughout the life of the living organism, or it occurs and stops somewhere?
It doesn't occur and stop anywhere. Since you are living, cells are being produced in you every now and then. It stops when you are dead.
Okay.
Yes, it stops when you are dead. So cells are being produced in you every now and then. And that immediately after. Are you married?
No, please.
You are not married yet. Yes. So immediately, let's say you give birth to one or two, it means that you cannot give birth with regards to the women or even with males after, like, let's say the honeybees, for instance. Males' organs are ripped off so that and the females will keep those organs together with the sex cells in their pouch for the rest of their lives. And so immediately they mate, they are dead and gone. Like we humans, even at the age of what, 90, a man can reproduce. A man can reproduce. Okay.
Yes, please.
But for women, and they have their cycles being altered, and that is why for males we don't have that problem so much. Okay. And that doesn't mean that women also don't have their DNA being replicated every now and then. So it's being produced day in, day out. Day in, day out. Is that right?
Thank you. Yes, please.
Very well. So, in review, our DNA replication occurs during the S phase of cell cycle. And you see, it all boils down to the mitosis and meiosis that we talked about earlier. I think it was in the first lecture that we discussed about that. Okay. Cell division. So it occurs during the S phase of cell cycle when chromosomes are in highly extended form. This type of replication is known as semi-conservative replication. And in that semi-replicative conservation, the two strands will separate from one another, but maintain their integrity, and each will synthesize the pool of nucleotides its complementary strand. So it produces a mirror copy of itself, and it continues. That is why, as a male, you wouldn't produce children who might not look like you. You produce children who would look like you, who have some semblance like you. Because DNA synthesis always proceeds in a 5-to-3 direction, and because the two DNA strands are arranged antiparallel with respect to each other, only one of the two newly synthesized strands can be made continuously. As the DNA away from the origin of replication, more DNA template is exposed. The other strand has to be made discontinuously in short pieces. So immediately leaves the nucleus into the cytoplasm to be breaking fragments. Because I'm free. I'm free. He's been imprisoned for a very long time in the nucleus. So immediately he's freed everywhere he wants to go. He goes to the nightclub. From the nightclub, he goes to the church. From the church, he goes to the mosque. From the mosque, he goes to the Buddhist temple. Everywhere. Okay, so they are exposed. So the other strand has to be made discontinuously in short pieces. And this later strand is called the lagging strand. The latter strand, the one that is behind in the nucleus, is referred to as what? The lagging strand. While the continuously synthesized strand is referred to as the leading strand. Yay! 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 It's free.
No, please,
The first-year students that has been given the opportunity to go home after the first semester, they are so happy to go as if they wouldn't come back. While continuously synthesized strand is called the leading strand. So the small pieces of DNA that comprise the lagging strand are called Okazaki fragment. And they are eventually aggregated together, forming a continuous DNA strand. So E. coli polymerase 3 initiates at the RNA primer, synthesizing DNA to fill in the gap up to the next RNA primer. However, it falls off the template because DNA, once it reaches the next RNA primer, at this point, DNA polymerase 1 takes over, and it contains a 5-to-3' exonuclease activity that can remove the RNA primer while simultaneously adds DNA nucleotides to the 3' end of the Okazaki fragment. DNA polymerase 1 cannot seal the gap between the two adjacent Okazaki fragments. This job is carried out by DNA ligase, which catalyzes the formation of a 3', 5' phosphodiester bond in an ATP-dependent reaction. So it takes one RNA polymerase, that's primates, two DNA polymerases, that's polymerases 2 and 1, and DNA ligase to synthesize the lagging strand. So once initiation occurs on the RNA primer, the leading strand only requires DNA polymerase 3 for its synthesis. So recently, the role of DNA polymerase E in DNA replication has been stressed upon so that the 3 DNA polymerases as alpha, gamma are now known to involve in eukaryotic DNA replication. DNA replication. So this ends session 1. So we are now moving on to unit 3, session 2. Unit 3, session 2, mutations in DNA. Mutations in DNA. And I'll give you just give me a minute. A minute. I'll be with you. I think I'm having some feedback here.
Hello.
All right. Hello.
Please, I wasn't hearing, so I thought I was so.
No, I had a technical hitch, so I just prompted the house. I'm back. So kindly mute. Thank you. Unit 3, session 2, and that has to do with mutations in DNA. So by the end of this session, to be able to state and explain the types of DNA mutations. And I believe you are taking notes. Then again, explain the causes of mutations, spontaneous mutations, error-prone replication, bypass errors, errors introduced during DNA repair, and induced mutations. What is mutation? If I may ask, what is mutation? It is the permanent alteration of the nucleotide sequence of the genome of an organism, virus, or extra chromosomal DNA, or other genetic elements. Permanent alteration. So, what is occurring in our waters, in the forest belt, in a few years to come? Humans with 8 limbs or 6 limbs will emerge from those areas. Why? Because their nucleotides have been altered permanently and to be carried on in subsequent generations. Mutation is a permanent alteration of nucleotide sequence of the genome of an organism, be it virus or extra chromosomal DNA or other genetic elements. So they result from errors during DNA replication or other types of damage to the DNA, which then they undergo error-prone repair or cause an error during other forms of repair, or else may cause an error during replication. Again, mutations may also
Hello, sir.
result from, yes.
With with the mutation, just as you said, it can occur either naturally or by induced method, just as you said with
Exactly.
our water body. Now, so I want to find out if it's an induced one, can't it be repaired? Hello, sir.
Yeah, I'm here.
Yeah, my question is, we know mutation is caused by two factors, either natural or by induced method. Now, my question is, with the induced method, can it be reversed? Can it be repaired?
It's so difficult repairing. It will be very difficult. Once you cause a blunder, it becomes very difficult to actually sort it out. You see the damage that the COVID caused. You saw the damage it caused, right? The vaccines that were produced, they were just some try and error things that they were doing. Initially, those who took it, some perished. That was why some people decided not to, get themselves involved in picking up those vaccines. Okay, so mutation, it's irreversible. It's so difficult. And there's no impossibility under the sun, but from where I sit, it is a no for me. Does that answer you?
Yes, partially, but I wanted to find out with the Galamsey situation that we find ourselves in our country, where all our water bodies are full of cyanide and other chemicals, which is causing a lot of mutations in features around western and other regions. If we stop this Galamsey and we give ourselves almost 20 years for this turbidity
20
level to go
years
down
is not
to
enough. 20 years is not enough.
clear the turbidity level of our
No,
water body.
you can't. It can't.
Wow.
It can't. It can't. Yeah, you should be looking at the next 400 to 800 years.
Hey!
So we are all dead.
And alive.
You are a dead man walking, just as I am.
In the name of Jesus, forgot it.
This is not in the name of Jesus.
No, sir.
So you see
sir,
the irreparable damage we are causing to our environment.
So,
you see the irreparable damage we are causing to our environment. And
yes,
you have learned people who would go to such an extent of even politicizing this deadly act. It's a gruesome. This is a genocide.
So, sir, that is where my argument is coming from. That we have intellectuals
Let's
who are defending these.
look at our mutation. If you are not careful, we will not continue the lecture. I beg you.
No, sir, because at the end of the day, we are all at stake.
Mm-hmm.
Our life is in danger. If we have people that are leading the front, championing this and supporting it, once they know the implication that is causing, and we still support it,
They are very much aware. They are very much aware. Mm-hmm. They are very much aware of the repercussions. They are very much aware of the repercussions. Mm-hmm. In opposition, I sing a different song. When I'm in authority, I dance to the tunes of my own drum. So let's continue. Let's pray God that everything. Let's continue. Let me continue, all right? So mutations may also result from insertion or deletion of segments of DNA due to mobile genetic elements. Then they may or may not produce discernible changes in the observable characteristics. Okay, they may or may not. Whilst you are eating the cocoyam, the cassava from those environments, instantly you wouldn't notice it, but it's a pile up. In some years to come, that is when you start experiencing organ failure and what have you. Nobody poisoned you, but it's as a result of the food chain. They are killing all ourselves gradually, because you don't know where the cassava is coming from. You don't know where the fish is coming from. All seeps into the sea. We go buy our fish, right? And we take it. Those fish contain cyanide, they contain arsenic, they contain mercury, and we are eating it. So the food chain is what? Poison. Mutations may or may not produce discernible changes in the observable characteristics of an organism. It can result in many different types of change in sequence. So please go download the film Wrong 10. Wrong 10. After this lecture, everybody should go and download the film Wrong 10. WRONG. Wrong 10. TURN. You have other episodes. I think Wrong 10, 1, 2, 3. Okay? Then you watch it. It's as a result of mutation that has taken place in some organisms. Some humans were living in a particular environment. They became mutated, and people got vanished. People who went on an expedition in a particular area were reported missing because they were being eaten by those individuals who had mutated. They became cannibals. So mutations in genes can have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. Due to the damaging effects that mutations can have on genes, organisms have mechanisms such as DNA repair to prevent or correct mutations by reverting the mutated sequence back to its original state. Causes of mutations. There are 4 classes of mutations that have been identified. And these are: 1, spontaneous mutations, molecular decay. Mutations due to error-prone replication, bypass of naturally occurring DNA damage, also called error-prone translation synthesis. Errors introduced during DNA repair induce mutations that are caused by what? Mutants. So scientists may also deliberately introduce mutant sequences through DNA manipulation for the sake of scientific experimentation. In 2017, a study claimed that 66% of cancer-causing mutations are random. This is 6%. So those of you who have been sleeping with your phones, please, from henceforth, stop sleeping with your phones. Switch them all off. There shouldn't be even radios and televisions in the rooms that you sleep. These are all causes of the cancers, rising cases of cancers that we have nowadays. We are exposing ourselves to lots of radiation. So a study in 2017 claimed that 66% of cancer-causing mutations are random. 29% are due to environment. And the study studied the population across 69 countries, and 5% are inherited. Just a few are able to pass it on to their children. 5% are passable. 69% is as a result of what the environment in which we live, environment we find ourselves. Like the most polluted place on earth. You know the most polluted place on earth?
No.
The most polluted place on earth was Agbogloshi, and it's still Agbogloshi in Accra, Ghana.
Chineke may God, we are dead.
You are not dead. We are living. We are existing. So let's continue. Now let's look at spontaneous mutation. That is one of the types of mutations. It occurs with non-zero probability. Yes, somebody is trying to reach. I was having a feedback. Oh, are you asking a question, please? Please mute for us, okay? Because you are giving us a feedback. So kindly mute for us. Thank you very much.
Hello, sir.
Let's look at. Yes.
Sir, some hands are up, but it seems you can't see them.
I can't see them. Oh, sorry.
Yeah, some hands are up. To Alex Opokwadman and Sabina, their hands are up. Maybe they want to ask a question.
Okay. Sabina. Sabina, please go ahead with your question. Okay, Alex.
Okay, look, please, I wanted to verify something there. We're talking about the movie Wrong Thing, and I wanted to know whether it's the American movie Wrong Thing.
Yes, please.
I have always thought it was a kind of a scary horror movie. So
yes,
that
is the horror. That is the horror associated with what is happening to our environment.
Oh, okay.
Yes.
Okay.
That is what we are creating for ourselves. You see how horrifying it is.
I just watched a trailer of it, so I couldn't watch again.
So that is the extent of damage that these so-called bourgeoisies are behind these galamseyers and they are doing to us. So you can imagine in the next 70 to 80 years to come, the sort of individuals will be breeding in this country. Can I continue, please?
And we allow somebody to leave his country and come and destroy it.
Please, let's talk about spontaneous mutation. Okay, let's drop the issue. I just educate you. So yours is to also go down and spread whatever you have learned. Exactly. We talk, they don't listen to us. So we are doing a bit. Spontaneous mutation. And it occurs with non-zero probability, even given a healthy, uncontaminated cell. They can be characterized by the specific change, and it includes tautomerism. Includes one, tautomerism. Okay? Tautomerism is a base changed by the repositioning of hydrogen atom, altering the hydrogen bonding pattern of that base, resulting in incorrect base pairing during replication. And we'll be looking at figure 2.1, which illustrates how a conversion from amino to imino form of cytosine changes, right? Instead of amino, amine. Amine has to do with what? Nitrogen, hydrogen forming what? Proteins. Instead of amino, there is an imino form of the cytosine changes the locations of hydrogen donor and acceptor groups, such that the imino form of cytosine base pairs with adenine instead of what? Guanine. Imino form of cytosine base pairs with adenine. C pairing with what? C pairing with what? A. And the normal is not supposed to pair. C is supposed to pair with G, guanine, right? But this one, because there is a mutation. Something has occurred as a result of what? An irregularity. So this will ultimately lead to a transition mutation. A transition mutation. So you realize that for the next couple of generations to come, would have deformities. And it will be normal because it will pertain for a very long time. Babies with 2 heads, 3 heads, and it will be normal because most of the inhabitants in that locality would bear those characteristics because their genes have been what? Altered. As for the other tautomers, the amino form of adenine looks sort of like a guanine and should be able to form 2 hydrogen bonds with cytosine. The enol form of guanine looks like an adenine. It should be able to form 3 hydrogen bonds with thymine. And the enol form of thymine looks like a cytosine and should be able to form 3 hydrogen bonds with guanine. So we'll be looking at figure 1, 2, 1, 2, 2, as well as 2, 3. And in all these cases, the wrong nucleotides will be inserted into the growing DNA chain. The normal, just like during the COVID times, the president was seen on air saying, we are not in normal times and will forever not be in normal times. After we being through that era, look at what is now happening to us. So here we go. The spontaneous mutations can be caused by tautomers. Tautomerism in cytosine. So you have adenine, original adenine, okay? And that is how it looks like. Then you have what? Cytosine. You have cytosine, okay? Cytosine and adenine. You realize that with adenine, you have how many you have a pentose sugar being attached to what? A hexose sugar, okay? 5-sided, pentagon, hexagon. But in cytosine, you have what? You have how many sides? A hexose sugar. The amino, okay, is a hexose sugar. 1, 2, 3, 4, 5, 6. But in adenine, you have a pentose and what? A hexose that are attached. Okay, now let's look at the imino as a result of what? Tautomerism, right? As a result of tautomerism. So you realize that the imine group has been replaced with regards to imino. The imine has been replaced. And you just have what? 1 imine forming the imino with regards to the adenine. Then the cytosine, the same applies to the cytosine. You realize that instead of the nitrogen, the double cycle being replaced with the hydrogen, you realize that it has a double cycle, or the double bond has moved. The double bond has moved from the 4th to the 3rd. So conversion from the amino, the imino form of what? Cytosine. Amino from the imino form of cytosine. And mutation caused by tautomer of cytosine. Okay. Hydrogen bond, normal tautomeric form, the cytosine. You have guanine. And the rare immunotautomeric form, right? So this is what mutagens causes there's a misrepresentation. The bonding is supposed to bond because it has no option. It has no option but to bond. Under normal circumstance, it wouldn't bond, but because there is no option, all the waters have been poisoned. ECG, Ghana water has no option than to what? To pump and use more chemicals in purifying the water. Under normal circumstance, those chemicals have a certain threshold that they are supposed to use. Now, instead of, let's say, 1 bag of the chemical they are supposed to be using, they are using about 20 to 30 bags to purify the water. I remember when I visited some parts for students supervision. When I came back, I've changed. And I said, oh, it's a result of the environment. He said, no, your skin has even changed because the water I used in bathing was something else. Although I wasn't experiencing it, but immediately I just came, they had seen the change. So you can imagine the damage that is being caused to individuals there. I was even drinking the water. And some of these individuals have no option but to drink it. So you can imagine the damage that is being caused as a result of these to merism. So the common cause, the common forms of guanine and thymine are the keto forms, which can rearrange to enol forms. So under normal circumstances, this is how guanine and thymine are supposed to what? To be arranged. Okay, but because of tautomerism, they rearrange in a certain mode and bring about chaos. So duprination, that's loss of a purine base, A to G, A or G, that is duprination. There's a loss of a base. So it has no option but that what? To pair like that. A is supposed to pair with T in DNA or pair with U in RNA. But here's the case, A is losing itself. So duprination is the loss of a purine base to form an apurinic site, AP site. Duprination. Deamination, hydrolysis, breaking of water changes a normal base to a typical base containing a keto group in the place of the original amine group. Examples include cytosine pairing to uracil and adenine to hypoxanthine, which can be corrected by DNA repair mechanisms. And 5-methyl cytosines pairing with what? Thiamine. Where from the 5-methyl cytosines? Because they have been altered. And the normal circumstance is supposed to be what? Methyl. And the normal circumstance is supposed to be what? Cytosine. But now a 5-methyl has been what? Attached to it. And it becomes 5-methyl cytosine instead of cytosine. So it has been mutated, which is less likely to be detected as a mutation because thiamine is a normal DNA base. So there are lots of people who have been mutated. You think this person is normal. He goes to church, he performs all other activities. But when he gets you, he eats flesh. That person has mutated and is now a cannibal. Slipped strand mispairing, denaturing of the new strand from the template during replication. followed by renaturation in a different spot. That is level, and this can lead to insertions or deletions. So when this occurs, when the mutations, spontaneous mutations occur, it's either delete certain nitrogenous bases or inserts what it likes. That's why some babies are being born with what, 2 heads. The left foot, the left limb, for instance, you would have another limb what germinating or protruding out of it. So Nipa Bakun has how many legs? 3, 4, 5. Well, what sort of human being? You have been mutated. So deamination of the 5-meter cytosine to produce thiamine. So you have cytosine, deamination occurring. Then to uracil, deamination of 5-meter cytosine cannot be repaired. So when the damage comes, per where I sit and per what I know, it cannot be repaired. It is irreversible. It cannot be repaired. Those scientists are of the view that for the next 500 to 800 years, that is when we'll be able to repair the damage. You can't repair it. 600 years, 500, where will we be? The damage will continue. Then they will even be using more sophisticated excavators for the mining. You see how even nowadays babies are playing with tablets. Even babies, they are tablets. Let's continue. Error-prone replication bypass. Although naturally occurring double-strand breaks occur at a relatively low frequency in DNA, their repair often causes mutation. So non-homologous end joining, HEJ, is a major pathway for repairing double-strand breaks. So the non-homologous end joining involves removal of a few nucleotides to allow somewhat inaccurate alignment of the 2 ends for rejoining, followed by addition of nucleotides to fill in gaps. So as a consequence of the non-homologous end joining, often introduces mutations. So for all those places that Galamsey is rife, right? For the next centuries to come, individuals from those environments will be mutated in individuals. So those who are involved in the activity itself, because they are exposing themselves to those chemicals. And mercury, even when you inhale it, the damage it causes to you as a human being is terrible. How much more you handling it? How much more you handling it? Look at the damage they have caused to the environment, to our water bodies, to our land, all in the name of gold. Induced mutation. We'll be able to talk about spontaneous mutation. We've done error-prone replication, and now on induced mutation. There are alterations in the gene after it has come into contact with mutagens and environmental causes. So induced mutations on the molecular level can be caused by chemicals such as hydroxylamine, base analogs, and DNA intercalating agents. Example, ethidium bromide, DNA cross-linkers, oxidative damage, and nitrous acid converts amine groups on the A and C to diazo groups, altering their hydrogen bonding patterns, which leads to incorrect or inaccurate base pairing during replication. So this is a typical example of the deamination by nitrous acid, cytosine, uracil, adenine, hypoxanthine, uracil, adenine, cytosine, hypoxanthine. When they pair up, oxidative damage, and nitrous acid converts amine groups on A and C. That's adenine and cytosine. The damage that is caused as a result of deamination by nitrous acid. So radiation, such as ultraviolet light, that's non-ionizing radiation, nucleotide bases in DNA, and that's cytosine and thymine, are most vulnerable to radiation that can change their properties. Okay? UV light can induce adjacent pyrimidine bases in a DNA strand to become covalently joined as a pyrimidine primer. So UV radiation in particular, possesses a longer wave. UV adenine can also cause oxidative damage to the DNA. So ionizing radiation, you ionize the radiation. Exposure to ionizing radiation, such as gamma radiation, can result in mutation, possibly resulting in cancer or death. So there are certain places which you get so much paid for a particular profession, but such professions you don't do for so long. Okay, you don't do for so long, such as places such as the Ghana Atomic Commission. So they are exposed to a lot of radiations. So the rippling effect is the possibility of either getting cancer or death in itself. So thymine dimer formation by
sir.
UV. Yes.
Sir, with the if you can go back to the ultraviolet radiation, I read it somewhere that at least even the sun rays can cause a bit of mutation on our system if you are exposed to it for a very long time.
Yes, it is. But we are lucky. We are lucky we have a lot of melanin in us, unlike the Scandinavians. Okay. They lack melanin. That is why when they come, most of them visit our beaches. They don't just come for the fact that, oh, Ghana is a nice place, and they come to heal themselves. Because there are certain places on earth that they barely experience enough sunshine.
So yes, sir, we having the melanin in our system and being exposed to it more. I don't want to make mention of other regions. Let's take the northern part of the country. When we are in the summer or the sunny time, you can see the sun there is very, very scorchy. Are they not at the risk of one point in their life being exposed to something of that nature?
Yes, the environment at certain points. That is why they have adopted certain lifestyles, like the hat. I remember the very first time I set foot way up north, I think about some 2 years back. Immediately got out. You see, it was an air-conditioned vehicle that I was traveling in. Immediately I got out, my eyes started watering because I wasn't used to the environment. It's as if somebody has whipped me and I'm crying. The heat that I came into contact with started making my eyes water. And I was told, oh, this is even cool. Around this time in Ouagadougou, you cannot stand it. Okay, so they have adopted certain lifestyles. So they put on the hat, right? they put on certain hats to minimize the extent of what? The heat that gets to them. In the deserts, they also come up with other modes of what? Livelihood. Is that right? So the UV, although it's good to an exposure, too much of everything is bad. Too much of everything is bad. That is why you normally have cases of meningitis being reported the northern and upper region, most especially in the upper regions down south. You barely hear cases of CSM, cerebrospinal meningitis, occurring down south. It occurs there because of the extreme temperatures, and that is when some of these organisms thrive. Does that answer your question, please?
Hello, sir.
Hello.
Doc. Please, I want to ask this question. Please, is it the presence of the melanin in our skin? Is it as a result of the sunlight presence, or is just by what means?
It is not by the sunlight. It is not by the sunlight. That's why when a Scandinavian gives birth here, they are not like us, but they are just like them. It's as a result of pigmentation. Okay. It's as a result of pigmentation. The same applies to you when you travel to another continent. Let's say in the European continent, or let's say in Asia, and you produce there. The fact that you are African doesn't mean that your progeny would be different because they are being produced in another environment. They will still be Africans.
Okay.
Is that right? So it's not as a result of where we find ourselves, but it's as a result of the pigmentation in us. We are lucky we have that. And unfortunately, some people, I don't know what is wrong with them, they take it off. They are taking it off. And when immediately you take it off, cancer is awaiting you. You want to become Obroni. While Obroni wants to become Bibini, but it's not finding the way. That's why he comes lying along our beaches. For lack of knowledge, my people perish. So mutations can be classified by effect on structure, effect on function, effect on fitness, impact on protein sequence, effect by inheritance. Our effect on structure. The sequence of a gene can be altered in a number of ways. Gene mutations have varying effects on health, depending on where they occur and whether they alter the function of essential proteins. Mutations in the structure of genes can be classified as small-scale mutations, large-scale mutations. Small-scale mutations include those affecting a small gene in one or a few nucleotides. And these include substitution mutations, often caused by chemicals or malfunction of DNA replication. Exchange a single nucleotide for another. These changes are classified as transitions or transversions. Then, exchanges a purine for a purine, or a pyrimidine for a pyrimidine. A transition can be caused by nitrous acid base mispairing or mutagenic base analogs such as BRD. Less common is a transversion, which exchanges a purine for a pyrimidine or pyrimidine for a purine. An example of a transversion is the conversion of adenine into cytosine. A point mutation are modifications of single base pairs of DNA or other small base pairs within a gene. So a point mutation can be reversed by another point mutation. A point mutation, it can be reversed, whereby in which the nucleotide is changed back to its original state. That's true reversion. Or by second site reversion, a complementary mutation elsewhere results in regained gene functionality. So humans over the years have evolved over the years, and our diet was initially it was plant-based before we became animal-based. We added, and now we've blended the two. We are now omnivorous, a bit of plant, a bit of animal. Point mutations that occur within the protein coding region of a gene may be classified into three kinds, depending upon what the urinary codons code for. So you realize that in our stomachs, in the alimentary canal, there's a point. There's a tip referred to as an appendix. An appendix.
Hello?
Hello, I'm not also hearing. I'm not seeing the screen. No audio.
May I
Is the
see the
network?
It's networking.
Is the network? So
Is it my side?
let's
Is
wait.
it my phone?
the network?
Really, only or crowd.
Let's go for break here. Fidelia is very
A
high.
human alimentary goes or heavy.
Oh, Mama Uncle break. Mama Uncle break. Mama Uncle break.
Oh, unless doctor comes.
I'm in break, oh, Mr. Poku.
Hey, since when?
I've break already about 5 minutes ago. I'm eating. Why?
Some of us don't take breakfast.
You
So
like
we
it.
take breakfast.
Mr. Opoku. Mr. Opoku.
Madam Juliet, we are on break, so we'll find something to eat. We shall be back.
Yes, Fidelia.
Who said it?
Please, at what time?
Who
What time
said
are we
it?
coming back?
Fidelia, you call. Please, nobody has given us break. Oh.
Come
If we
on.
come back
We
and
are
we're
not
not
on
giving,
break.
I
I'm
have
Come say
break.
you
talking
are
to
not
him.
on break.
Let me finish talking to him. I'll get back to you, please.
Okay, but I'm on break, so
Thank you. You can
You
enjoy
are
your
on
break.
break. The class is not on break.
no problem.
Hello. Please, we are going for officially we are on break now. We are returning 9:40. 20 minutes break. We are coming back 9:40.
Yeah, that's good.
This is from
Let's hope.
Dr. So you can go and find something. For those who have already eaten, relax. We are coming back exactly 9:40. You know, doctor does not waste his time.
All right. Thank you.
Thank you.
Hello.
Stop it. Oh no.
full delay karma pelimbo. The guy is smart. What apart from stock that was that one. I'm going to start it. We are going to start it. And open, yeah, man, not you could post Hello. Hello.
Hello.
Hi.
What is happening?
What is happening as in what? Doctor is joining, please. So let's get prepared. Just mute yourself. You'll soon join us. Yeah, so Doc, we got to this place, effect on structure.
All right, welcome back. Sorry for the long break. It was in your good. Types of mutations can be classified by effect on structure, effect on function, effect on fitness, impact on protein sequence, and effects by inheritance. So let's pick them one after the other and digress. So effect on structure, the sequence of a gene can be altered in a number of ways, as gene mutations have varying effects on health, depending on where they occur and whether they alter the function of essential proteins. So that's why the rising cases of cancer, I've told you not to be sleeping with phones, not to be sleeping in rooms with TVs on and what have you. Mutations in the structure of genes can be classified as small scale mutations and large scale mutations. So even with mutation, we have small scale as well as large scale mutations. So the small scale mutations include those affecting a small gene in one or a few nucleotides. And it includes substitution mutations, often caused by chemicals or malfunction of DNA. Substitution mutations. So that is what is going to occur in those areas that have been poisoned with arsenic, cyanide, and mercury. So exchange of a single nucleotide. That's a malfunction of a DNA replication, exchange a single nucleotide for another. And these changes are classified as transitions or transversions, as we looked at in the other slide. So most common is a transition that exchanges a purine for a purine, adenine for one, or pyrimidine for pyrimidine, that's cytosine for thymine, instead of the other way around. So a transition can be caused by nitrous acid base mispairings or mutagenic base analogs such as BRDU. So less common is a transversion, which exchanges a purine for a pyrimidine. I think I've gone back, but don't worry. A pyrimidine for a purine. An example of a transversion is a conversion of adenine into cytosine, a point mutation where modifications of a single base pairs of DNA or other small base pairs in a gene. So a point mutation can be reversed by another point mutation in which the nucleotide is changed back to its original state. That's true reversion. Or by second site reversion, a complementary mutation elsewhere that results in regained gene functionality. The point mutations that occur within the protein coding region of a gene may be classified into three kinds, depending upon what the erroneous codons code for. And we talked on silent mutations, which codes for the same amino acid, missense mutations, which code for a different amino acid, nonsense mutation, which also codes for a stop codon and truncate protein. So insertions add one or more extra nucleotides into the DNA, and they are usually caused by transposable elements or errors during replication of repeating elements. Deletions remove, because you're deleting, it removes one or more nucleotides from the DNA, like insertions, and you putting in certain codons. So these mutations can alter the reading frame of the gene. So in general, they are irreversible. So we looked at this diagram area where there's a transition from a thymine to an adenine, cytosine to a guanine, where it was replaced. Okay, transition, it was transversed. So initially it was up, now it is down, and there's a deletion. It is deleted. It's deleted from that stage, and there's an insertion. Insertion again. We initially started with the transition was C to G, and now the transversion has been inserted. So these large scale mutations, this has to do with small scale mutations. Now let's look at the large scale mutations in chromosomal structure. And they include amplifications. Amplify. You know, when you go to church, you go to the mosque, and they are using the microphones. They need to amplify their voices. So amplification or gene duplications leading to multiple copies of all chromosomal regions. Leading to multiple copies of all chromosomal regions. So it increases the dosage of the genes located within them. Then deletions. So the mutations include amplifications, deletions, and mutations. So deletions of large chromosomal regions leading to loss of the genes within the regions. So just like the figure that we just observed. Then mutations, whose effect is to suppose previously separate pieces of DNA are potentially bringing together separate genes to form functionally distinct fusion genes. Right? So it includes the following: chromosomal translocation, interchange of genetic parts from non-homologous chromosomes, those which are not similar, non-homologous chromosomes. For instance, somebody has a hand, then all of a sudden, instead of the eyes being fixed on the head, the person's hands will be bearing what? The eyes. Interchange of genetic parts from non-homologous chromosomes. Then interstitial deletions, and intrachromosomal deletion that removes a segment. For example, cells isolated from a human astrocytoma, a type of brain tumor, were found to have a chromosomal deletion, removing sequences between fused in glioblastoma gene and the receptor tyrosine kinase produce protein. So the abnormal fused in glioblastoma and that of tyrosine receptor tyrosine kinase protein has constitutively active kinase activity that causes oncogenic transformation from normal cells to what? Cancer cells. Why is it that lots of people are saying if we work in the Galamsey site, we will die early, we will develop cancer, and we are not getting cancer? It doesn't happen instantly. It takes time. And after the next 5-10 years, that's why those miners who are working with Anglo Gold, the maximum number of years a miner works in a particular part, for instance, a mine, and it's about some 10, maximum 15, 20 years. They retire and take off benefits, because when you are exposed to the chemicals for a very long time, all you do is you pick the money and you go and give somebody to spend it for you. You might have developed lots of cancer cells. So chromosomal inversions, reversing the orientation of a chromosomal segment. And again, loss of heterozygosity, loss of one allele, either by deletion or genetic recombination event in an organism that previously had two different alleles. So to change the genetic makeup of that individual, change the genetic makeup of that individual. So in some years to come, those coming from those areas, you realize that their genetic makeup will be what? Thoroughly changed. Effect on function. They include loss of function, mutations, function of gain of function mutations, and dominant negative mutations. So loss of function mutations, also called inactivating mutations, result in the gene product having less or no function. That's impartially or wholly inactivated. So when the allele has a complete loss of function, or allele, it is often called an amorph or amorphic mutation in the Miller's morph schema. Phenotypes associated with such mutations are most often recessive. Exceptions are when the organism is haploid or when the reduced dosage of a normal gene product is not enough for a normal phenotype, and this is called haploinsufficiency. So gain of function mutations, also called activating mutations, change the gene product such that its effects get stronger, enhance activation, or even is superseded by different and abnormal function. When the new allele is created, heterozygote containing the newly created allele, as well as the original, will express the new allele genetically. This defines the mutations as dominant phenotypes. And several of Muller's morphs correspond to gain of function, including hypermorph and neuromorph. Dominant negative mutations, also called antimorphic mutations, have an altered gene product that acts antagonistically to the wild type allele. So these mutations usually result in an altered molecular function, often inactive, and are characterized by a dominant or semi-dominant phenotype. So in humans, for instance, dominant negative mutations have been implicated in cancer. Mutations in genes P53, ATM, CBAPA, and PPAR gamma. Marfan syndrome is caused by mutations in the FB and I gene. These are all type of cancers located on chromosome 15, which encodes fibrin 1, a glycoprotein component of the extracellular matrix. Marfan syndrome is also an example of dominant negative mutation and haploinsufficiency. Effects on fitness. We've done effects on function, so we are now on effect of fitness. So in applied molecular genetics, it is usual to speak of mutations as either harmful or beneficial. So a harmful or deleterious, or harmful or deleterious, some are called deleterious or harmful mutation. It decreases the fitness of the organism because it is harmful, it is harming the organism. So a beneficial or advantageous mutation increases the fitness of the organism. Mutations that promote traits that are desirable are also called beneficial. So I remember growing up. Yes.
Can we have a typical example of a advantageous mutation where it has
Yes,
enhanced?
that was what I was about saying, and you just interjected. When growing up, there was a movie on our scenes, maybe the lightning, and there are certain fishes that have electric waves that they use to, hunt and guard themselves. Some eels, some fishes, they contain a lot of very powerful hydroelectric energy. When you touch them, it will electrocute you and you might die. Okay, and it's rare in humans. And there was this couple. The male, I think, was working in a reductive area, and I think was exposed to some form of radiation. So when they gave birth to their son, that boy was possessed with wonderful powers. He could electrocute you. Okay, he could electrocute you. So to him, it was advantageous. But it has its negative tendencies. it had its negative tendencies. So there are lots of beneficial mutations occurring with regards to humans. So a beneficial advantageous mutation increases the fitness of the organism. It gives you an added advantage over the normal human being. So somebody misbehaved in public and you just stretch your hand and you have electrocuted that person. You've not shot the person. The police can come in, they can do whatever, but you didn't use any weapon. So it's advantageous to that individual, but to an extent, everything has its pros and cons. So in theoretical population genetics, it is more usual to speak of mutations as deleterious or advantageous than harmful or beneficial. We normally use deleterious or advantageous than using harmful or beneficial. A neutral mutation has no harmful or beneficial effect on the organism, and such occur at a steady rate. It's slow-paced, forming bases for the molecular clock. So, in neutral theory of molecular evolution, neutral mutations provide genetic drift as a basis for most variation at the molecular level. A nearly neutral mutation is a mutation that may be slightly deleterious or advantageous, although most nearly neutral mutations are slightly deleterious. Impact on protein sequence. So a frame shift mutation is a mutation caused by insertion or deletion of a number of nucleotides that is not evenly visible by 3 from a DNA sequence. So due to the triplet nature of gene expression by codons, the insertion or deletion can disrupt the reading frame or the grouping of the codons, resulting in a completely different translation from the original. So the earlier in the sequence the deletion or insertion occurs, the more altered the protein produces. So for example, the code CCU, GAC, UAC, and CUA codes for the amino acid protein aspartic, tyrosine, and leucine. So if the U in CCU was deleted, the resulting sequence would be CCG, ACU, ACC, UA, which would instead code for proline, threonine, and threonine. In contrast, any insertion or deletion that is evenly divisible by 3 is termed in frame mutation, is termed an in-frame mutation. So a nonsense mutation is a point mutation in a sequence of DNA that results in a premature stop codon or a nonsense codon in the transcribed messenger RNA, and possibly a truncated and often non-functional protein product. So this sort of mutation has been linked to different mutations such as congenital adrenal hyplasia. It's a condition. It's a condition that normally affects the brain. Missense mutations or non-synonymous mutations are types of point mutations where a single nucleotide is changed to cause substitution of a different amino acid. So this in turn can render the resulting protein non-functional. Such mutations are responsible for diseases such as epidermolysis bullosa, sickle cell disease, and SOD1 mediated ALS. So effect by inheritance. In multicellular organisms, with dedicated reproductive cells, mutations can be subdivided into germline mutations, which can be passed on to descendants through their reproductive cells, and somatic mutations, also called acquired mutations, which involve cells outside their dedicated reproductive group and which are not usually transmitted to descendants. A germline mutation gives rise to a constitutional mutation in the offspring, that is, a mutation that is present in every cell A constitutional mutation can also occur very soon after fertilization or continue from a previous constitutional mutation in a parent. So the distinction between germline and somatic mutations is important in animals that have a dedicated germline to produce reproductive cells. However, it is of little value in understanding the effects of mutations in plants, which lack dedicated germline. The distinction is also there in those animals that reproduce asexually through mechanisms such as budding, because the cells that give rise to the daughter organisms also give rise to that organism's germline. A new germline mutation that was not inherited from either parent is called a de novo mutations. A de novo mutation. So diploid organisms, humans, for instance, contain 2 copies of each gene, a paternal and a maternal allele. You pick a little, 50% from mother, 50% from mother, and that forms your genes. Based on the occurrence of mutation on each chromosome, we may classify mutations into 3 types: heterozygous, homozygous, and compound heterozygous. A heterozygous mutation is a mutation of only one allele. Mutation of only one allele. Homozygous mutation is an identical mutation of both the paternal and maternal alleles. I think the mix up here, it should be the other way around. It's a mutation of only one allele because it is homo, it's one, but heterozygosity is rather the combination of mother and father. Compound heterozygous mutations or genetic compound consist of 2 different mutations in the paternal and maternal alleles. So in review, mutation can result in many different types of change in DNA sequences. Again, mutations in genes can either have no effect, alter the product of a gene, or prevent a gene from functioning properly or completely. It is the permanent alteration of the nucleotide sequence of the genome of an organism, virus, or extra chromosomal DNA or other genetic elements. The mutations result from errors during DNA replication or other types of damage to DNA, which may undergo error-prone repair or cause an error during other forms of repair, or else may cause an error during replication. So classes of mutation have also been identified, and these are the spontaneous mutations as molecular decay mutations due to error-prone replication, bypass of naturally occurring DNA damage, also called error-prone translesion synthesis, errors introduced during DNA repair, and induced mutations caused by mutagens. And finally, mutation types could be classified by the effect on structure, function, fitness, and impact on protein sequence, and effect by inheritance. So that brings us to the end of Unit 3, Session 2. We are now moving to Session 3, that is DNA repair. So by the end of this, you should be able to discuss DNA repair mechanisms and explain how DNA repair affects aging. Again, relate some hereditary DNA disorders to the ability of DNA to repair. So DNA repair mechanisms. It results in physical abnormalities in the DNA, such as single and double strand breaks, 8-hydroxydeoxyguanosine residues, 8-hydroxydeoxyguanosine residues, and polycyclic aromatic hydrocarbon adducts. So DNA damage can be recognized by enzymes. DNA that have been damaged, recognized by enzymes, and thus can be correctly repaired if redundant information, such as the undamaged sequence in the complementary DNA strand or in a homologous chromosome, is available for copy. If it is readily available in the nucleus, it will quickly repair itself. If a cell contains DNA damage, transcription of a gene can be prevented, and thus translation into a protein will also be blocked. Replication may also be blocked, or the cell may die. In contrast to DNA damage, a mutation is a change in the base sequence of the DNA, and it cannot be recognized by enzymes once the base change is present in both DNA strands, and thus a mutation cannot be repaired. So mutations are irreversible. So at the cellular level, mutations can cause alterations in protein function and regulation. Mutations are replicated when the cell replicates. In a population of cells, mutant cells will increase or decrease in frequency according to the effects of the mutation on the ability of the cell to survive and reproduce. Cells cannot function if DNA damage corrupts the integrity and accessibility of essential information in the genome, but cells remain superficially functional when non-essential genes are missing or damaged. Depending on the type of damage inflicted on the DNA's double helical structure, a variety of repair strategies have evolved to restore lost information. So if possible, cells use the unmodified complementary strand of the DNA or sister chromatid as a template to recover the original information. So without access to a template, cells use an error-prone recovery mechanism known as translation synthesis as the last result. And that is what is going to happen in the areas mentioned earlier, where mining is rife now. Damage to DNA alters spatial configuration of the helix, and such alterations can be detected by the cell. Again, once a damage is localized, specific DNA repair molecules bind at or near the site of damage so that they can quickly resolve it, inducing other molecules to bind and form a complex that enables the actual repair to take place. So the body in itself heals itself to an extent. So it isn't all diseases that you need medication to. So at times, just leave the body. It will sort out itself. So the three general mechanisms of discussion include direct reversal. Direct reversal, it occurs. Double strand breaks and translation synthesis. So looking at that. Direct reversal mechanism. Direct reversal mechanism. So cells are known to eliminate three types damage to their DNA by chemically reversing it. And these mechanisms do not require a template, since the types of damage they contract can occur in only one of the four bases. So such direct reversals are specific to the type of damage incurred and do not involve breakage of the phosphodiester backbone. So the following discusses the three types of direct reversal mechanisms. The first is base excision repair. Base excision repair, BR, repairs damage to a single nitrogenous base, and the nitrogenous bases are the adenine, guanine, just one. Base excision repair. So it repairs damage to a single nitrogenous base by deploying enzymes called glycolysis. So these enzymes remove a single nitrogenous base, to create an epurinic or epimenidic site, EP site, epurinic or epurimidic site, as in purine and pyrimidine. Epurinic, epurimidic site, AP sites. So the enzymes called AP endonucleases, they nick the damaged DNA backbone at the AP sites. Apurinic. They are not purines. They are not pyrimidines, but they bear copies of it. Okay, they are not real purines. They are not real pyrimidines. Apurinic, and pyrimidinic. Okay, so the AP site. So the DNA polymerase then removes a damaged region using its 5' to 3' exonucleases activity and correctly synthesizes a new strand using the complementary strand as a template. So the nucleotide excision repair, that has to do with the base repair. So this is the nucleotide excision repair. And it repairs damaged DNA, which commonly consists of bulky helix distorting damage. The bulky helix, the double-stranded helix, okay? Bulky helix distorting damage, such as pyrimidine dimerization caused by UV light. Pyrimidine dimerization caused by UV light. So the damaged regions are removed in 2 to 12 nucleotide long strands in a 3-step process, which consists of recognition of the damage, excision of the damaged DNA, both upstream and downstream of the damage by endonucleases, and the resynthesis of the removed DNA region. So that is what it actually does with regards to repairing a nucleotide. So this is what happens in the nucleotide excision repair. So in the excision repair, you realize there are 2 diagrams: base excision and nucleotide excision. So with the base, you realize deamination, ATG, CUG, CAT, and TGA. And uracil is replaced. The uracil, this as occurring on an RNA. Uracil DNA glycolysis. So it is replaced. Then the repair nucleases, you realize that most of that part goes off. Then DNA polymerase beta acts on it and replaces it with what? GCC instead of what? The BCU. So there is a DNA ligase occurring, base excision repair. It works on just then the thymine dimer, X nuclease. You have a TT which goes into an AT and an AG in the nucleotides, and that has to be within the range of 30 nucleotides. Okay, so the DNA polymerase beta, okay, replaces it in that fashion: GCC, GCA, ATG, AT, right? And that informs the nucleotide excision repair. Illustration of how the base nucleotide excision repairs takes place. Mismatch repair. Mismatch repair. This type of repairs is present in essentially all cells to correct errors that are not corrected by proofreading. Mismatch. This type of repair is present in all cells to correct errors that are not corrected by proofreading. There are some that it's as an oversight. You barely see it, but the cell and the enzymes will actually form ways to repair them. These systems consist of at least 2 proteins. One detects the mismatch, the other recruits endonucleases that cleave the newly synthesized DNA strand close to the region of damage. So in E. coli, proteins involved are the MUT class proteins, and it's followed by removal of damaged region by the exonucleases. There are two types of nucleases we talked about last week, endo as well as exonucleases. Then resynthesis by DNA polymerase and next sealing by DNA ligase. So mismatch, post replication repair. So the parental strands are methylated. You apply methylated spirits to the parental DNA on certain adenine bases. Okay, so you have methane, methyl, CH3, okay, on the 5' and on the 3'. One moving inwards, the other moving outwards. Lagging, one is lagging and the other is leading. So mutations on the newly replicated strand are identified by scanning for mismatches prior to methylation of the newly replicated DNA. So the mutations are repaired by excision repair mechanisms. Then after repair, the newly replicated strand is methylated. The new one that has been repaired is methylated. And maybe continue your research or experiment on the other hand. Double strand breaks. Double strand breaks in which both strands in the double helix are severed. Both strands are severed. Maybe something occurred and there might be breaks. They are particularly hazardous to the cell because they can lead to genome rearrangements. It is known that double strand breaks and a cross linkage joining both strands at the same point is irreparable because neither strand can then serve as a template for repair. The cell will die in the next mitosis or in some rare instances, mutate. 3 mechanisms exist to repair double strand breaks. 1. Non-homologous end joining, NHEJ. Microhomology mediated end joining, and homologous recombination. These are the 3 mechanisms that exist to repair the double strand breaks. Non-homologous end joining, microhomology mediated end joining, and the homologous recombination. So an in vitro simply means something which is ongoing, it's introduced, right? An in vitro system occurred in mammalian cells at the levels of 20% of homologous recombination, and both homologous recombination and non-homologous end joining mechanisms were also available. So the double strand breaks. Homologous recombination requires the presence of an identical, because they are homologous, like identical twins. They look alike. If you are not careful, you might not see the first from the second if you are not very close to them. Homologous recombination requires the presence of an identical or nearly identical sequence to be used as a template for repair of the break. So enzymatic machinery responsible for this repair process is nearly identical to the machinery responsible for chromosomal crossover during meiosis. This pathway allows a damaged chromosome to be repaired using a sister chromatid that's available in G2 after DNA replication, or a homologous chromosome as a template. Double strand breaks caused by the replication machinery attempting to synthesize across a single strand break or unrepaired lesion cause collapse, replication fork, and are typically repaired by recombination. So the extremophile Deonococcus radiodurans has a remarkable ability to survive DNA damage. The extremophile Yes.
So please, what is this long thing? Is it an enzyme, a viral, or is just a part of the DNA
It is
strand?
part of the radioactive element that extremophile. Okay, it is part of the radioactive element used in breaking the DNA. So it has a remarkable ability to survive DNA damage from ionizing radiation and other sources. So at least 2 copies of the genome with random DNA breaks can form DNA fragments through annealing. Annealing is purging, putting up together these strands. So partially overlapping fragments are then used for synthesis, for the process of the homologous regions through a moving D loop. It acts in a loop, like hovering. Let's say if it's round, it's like a car moving round, entering a roundabout. But this one, it moves in a D fashion. So it moves through a D loop that can continue extension until they find complementary partner strands. So the final step is a crossover by means of Rec-A dependent homologous recombination, recognizable adenine dependent homologous recombination. So the top isomers introduced. Isomers are either chemicals or enzymes, which bears or is a mirror copy of the original. Top isomerases introduce both single and double strand breaks, the cause of changing the DNA state of supercoiling, which is especially common in regions near an open replication form. Such breaks are not considered DNA damage because they are a natural intermediate in the top isomerase biochemical mechanism and are immediately repaired by the enzymes that created them. Translation synthesis. It is a DNA damage tolerance that allows the DNA replication machinery to replicate past DNA genes, such as thiamine dimers. So it involves switching out regular DNA polymerases for specialized translation polymerase. That is DNA polymerase 4 and 5 or 5 from the Y polymerase family. So earlier made emphasis of the fact that there are 10 subunits of these polymerases, okay, often with larger active sites that can facilitate the insertion of bases opposite damaged nucleotides. So the polymerase switching is thought to be mediated by, among other factors, the post-translational modification of the replication processivity factor, PCNA. Translation synthesis polymerases often have low fidelity, that's high propensity to insert wrong bases. So they are within the nucleus and they stand a very high chance of introducing the wrong codons. So they have a high affinity or propensity to insert wrong bases on damaged templates relative to regular polymerases. So many are extremely efficient at inserting correct base opposite specific types of damage. DNA repair and aging. DNA repair and aging. So DNA repair rate is an important determinant of cell pathology. Experimental animals with genetic deficiencies in DNA repair often show decreased lifespan. If the rate of DNA damage exceeds the capacity of the cell to repair it, the accumulation of errors can overwhelm the cell and result in early senescence, apoptosis, or cancer. Inherited diseases associated with faulty DNA repair functioning result in premature aging. And some people, when you see them, you think you have seen your grandfather. Well, that person is very young. It's as a result of these inherited traits from the family ancestry. Increased sensitivity to carcinogens. Increased sensitivity to carcinogens, corresponding increased cancer risk. So on the other hand, organisms with enhanced DNA repair systems, such as radioendurance, radioendurance, the most radiation-resistant known organisms, exhibit remarkable resistance to the double-strand break-inducing effects of radioactivity, likely due to enhanced efficiency of DNA repair. So some people with their immunity, immediately you expose them to some of these radioactive elements, they begin growing some cancer cells. But others too, because their DNAs are so potent, they are able to do away with some of these carcinogens in their systems, and they are ready to survive. So that actually helps in DNA repair as well as aging. Medicine and DNA repair modulation. So defects in the nucleotide excision repair mechanism are responsible for several genetic disorders. It include xeroderma pigmentoso. Such individuals, xeroderma, as in zero skin pigment, hypersensitivity to what? Sunlight or UV, resulting in increased skin cancer incidence and premature aging cocaine syndrome. Premature aging cocaine syndrome, hypersensitivity to UV and chemical agents. Some people have very, very sensitive skin. Some people are very, very sensitive. We are very lucky to possess a lot of melanin. We are very lucky to possess a lot of melanin, though it is not so prominent in our environment like in the Scandinavian areas or the temperate areas. Over there, they do not have enough UV exposure, so they come up with some of these disorders.
Hello, sir.
Zero pigmentoso, right?
Yes, sir. Hello, sir.
They are very sensitive. Yes.
Yes, I wanted to find out this xerodermapigmentosium.
Yeah, pigmentoso.
Yes, in our part, can we refer to that as the albinos that we have around?
To some extent.
Yes, because I have one albino within my area here. I have one albino within my area here. When the sun is out, he can't step out. You always have to be indoors. Or even when he's stepping, I have to use, say, because something, the ladies can help me, say sunshade or what, some cream like on the body. And if you get finished,
yes,
the skin begins to peel, like you can hold the skin and be peeling the skin off.
Exactly. And when they are aging, it is terrible with regards to
very
the skin.
Good. Yes. He's around 30 and then he cannot stand the sun at all.
It's a disorder.
Okay, thank you very
It's
much.
a disorder. It's a genetic disorder, right? The next is trichodystrophy. Trichodystrophy. This is sensitive skin. They have brittle hair and nails. And when we have Werner syndrome, that's premature aging and retarded growth. There are some people who have this deformity. Okay, they are very old, like those people that are being used in our movies nowadays. These Lilliputians, like Double and
okay,
yes, them kids. Okay, Double and those other individuals. All right, they have premature aging, retarded growth. You see that he's old. YOC and the rest, right? He's an old man. He's old, but it looks as if he's a very young person. Okay, these are all DNA modulations. Then Bloom syndrome, sunlight and hypersensitivity, high incidence of malignancies, especially leukemia. We have cancerous blood systems, and they don't last. Immediately you get leukemia, about saying bye-bye to this world. No amount of therapy can sustain you. You eventually pass on. And ataxia telangiectasia, ataxia, telangiectasia. These are sensitive to ionizing radiation and chemical agents. Most of these things, luckily for us, and unfortunately for us, we don't have so much in our regions. Okay, it's normally associated with outside world, the temperate and more advanced nations, because they actually research into the highest levels of atomicity and what have you. So it is very common in those environments than we in the tropics, in developing countries. All of these diseases are often called segmental progerias or accelerated aging diseases because victims appear elderly and suffer from aging-related diseases at an abnormally young age, while not manifesting all the symptoms of old age. And other diseases associated with reduced DNA repair function include fanconi-anemia. These are hereditary breast cancer and hereditary colon cancer diseases. Fanconi-anemia. So repair and cancer. Because of inherent limitations in the DNA repair mechanisms, if humans lived long enough, we would all eventually develop cancer. That's according to somebody's research. You can disbank it or you can align with it. There are at least 34 inherited human DNA repair gene mutations that increase cancer risk. And many of these mutations cause DNA repair to be less effective than normal. So in particular, hereditary non-polyposis colorectal cancer is strongly associated with specific mutations in the DNA mismatch repair pathway. So BCRA1 and BCRA2, these are two famous genes whose mutations confer a hugely increased risk of breast cancer. Sorry, it is BCRE1 and BCRE2. A hugely increased risk of breast cancer on carriers are both associated with a large number of DNA repair pathways, especially non-homologous joining and homologous recombination. So the basic processes of DNA repair are highly conserved among both prokaryotes as well as eukaryotes, even among bacteriophages. However, more complex organisms with more complex genomes have correspondingly more complex repair mechanisms. The ability of a large number of protein structural motifs to catalyze relevant chemical reactions has played a significant role in the elaboration of repair mechanisms during evolution. So an extremely detailed review of the hypothesis relating to the evolution of DNA repair. So fossil records indicates that single cell life began to proliferate on the planet at some point during the Precambrian period. And these are years, medieval years, billions of years ago. Although exactly when recognizable modern life first emerged is unclear. So nucleic acids become the sole and universal means of encoding genetic information, requiring DNA repair mechanisms that in their basic form have been inherited by all extant life forms from their common ancestor. So the emergence of the Earth's oxygen-rich atmosphere, known as oxygen catastrophe, due to photosynthetic organisms, as well as the presence of potentially damaging free radicals in the cell due to oxidative phosphorylation, necessitated the evolution of DNA repair mechanisms that act specifically to counter the types of damage induced by oxidative stress. So, on some occasions, DNA damage is not repaired or is repaired by an error-prone mechanism that results in a change from the original sequence. When this occurs, mutations may propagate into the genome of the cell's progeny. So you change. Should such an event occur, should such an event occur in a germline cell that will eventually produce a gamete, the mutation has the potential to be passed on to the organism's offspring. The rate of evolution in a particular species or in a particular gene is a function of the rate of mutation. So as a consequence, the rate and accuracy of DNA repair mechanisms have an influence over the process of evolutionary change. And since the normal adaptations of population of organisms to changing circumstances, for instance, the adaptation of the breaks of a population of finches to the changing presence of hard seeds or insects, proceeds by gene regulation and the combination and selection of gene variations, not by passing of irreparable DNA damages to the offspring. So, in review, a mutation cannot be recognized by enzymes once the base change is present in both strands, DNA strands, and as a mutation cannot be repaired at the cellular level. Mutations can cause alterations in protein function regulation. At the cellular level, mutations can cause alterations in protein function and regulation. Mutations are replicated when the cell replicates. Damage to DNA can't be recognized by enzymes that could be correctly repaired if the redundant information, such as the undamaged sequence, the complementary DNA strand, or in a homologous chromosome, is available for copy. So, if a cell retains DNA damage, transcription of a gene can be prevented. Consequently, block translation into the protein. The great majority of mutations that are not neutral in their effect, are deleterious to a cell's survival. Thus, in a population of cells composing a tissue with replicating cells, mutant cells will tend to be lost. However, infrequent mutations that provide a survival advantage will tend to clonally expand at the expense of neighboring cells in the tissue. They will clone, they will form new ones, depicting the replica. So this advantage to the cell is disadvantageous to the whole organism because such mutant cells can give rise to cancer. So immediately they start forming, they start forming in classes. That's why they are being told to watch your diet, especially those cancerous patients. They watch their diets. It's not everything that they take, because immediately they take in something, the diet could replicate the growth of those cancerous cells. So the three general mechanisms by which DNA can be repaired include direct reversal, double-strand breaks, and translation synthesis. Examples of direct reversal repairs are base excision repair, nucleotide excision repair, and mismatch repair. Experimental animals with genetic deficiencies in DNA repair exhibit decreased lifespan. Inherited diseases associated with faulty DNA repair functioning result in premature aging. It increased sensitivity to carcinogens and correspondingly increased cancer risk. Again, defects in the nucleotide excision repair mechanism are responsible for several genetic disorders. The basic processes of DNA repair are highly conserved among both prokaryotes and eukaryotes, and even among bacteriophages. So that brings us to the end of Unit 3, Session 3. Unit 3, Session 4, defects in DNA repair and replication. So by the end, you should understand how the defects in DNA repair and replication result in some diseases. And again, explain how some human genetic diseases are linked up to cancer. Explain how some of these diseases are linked up to cancer. Nucleotide excision repair syndromes. So lesions that cause gross distortions on the DNA double helix, such as pyrimidine dimers induced by UV irradiation, are recognized and excised from DNA by a complex mechanism known as nucleotide excision repair. Nucleotide excision repair. So three human genetic disorders associated with defects of UV nucleotide excision repair include the xerodermapigmentosum, XP, cocaine syndrome, CS, and trichothiodystrophy, TTD, XP, CS, TTD. So we have a summary of this in a table that has been generated for you. Human syndromes affected in nucleotide excision repair. Then xerodermapigmentosum. Right? In main clinical features, sunlight hypersensitivity, you have high incidence of skin cancer, frequent neurological abnormalities, and it occurs in what? Premature aging. In the cellular characteristics of xerodermapigmentosum XP, hypersensitive to and hypermutable by UV and some chemical agents. 8 complementation groups, XPA to XPG and XPV. Some patients are affected in transcription, coupled repair, in overall repair, or both. Pain syndrome, sun sensitivity, mental retardation, that is microcephaly, microcephaly. Then dwarfism. Normally, no cancer is associated with the cocaine syndrome. Hypersensitivity with regards to cellular characteristics. Hypersensitivity to UV and chemical agents, right, such as colchicine. Then defective transcription after UV. At least some patients have defective transcription coupled repair to oxidative induced damage. Then you have trichothiodystrophy, TTD. Sensitive skin with regards to the main clinical features. They have sensitive skin, they have brittle hair and nails. Then they also possess frequent physical and mental retardation. Normally, no cancer is associated. Not so common in our environment. Defective repair with regards to its cellular characteristics. Defective repair. Gray complementation groups, including TTD, not cloned. You cannot clone them because they are not generative. The normal immunological responses to UV, contrary to XP patients, contrary to the zero pigmentoso patients, with mutations in the same gene. Then nucleotide excision repair syndromes. Individuals are highly sensitive, representing dramatic pigmentary skin disturbances, especially at regions exposed to sunlight. Then again, there is a great increase in the incidence of both benign and malignant skin tumors. There is great increase in the incidence of benign and malignant skin tumors. And frequently, these clinical features are accompanied by neurological abnormalities and premature aging. So fibroblasts from these patients are sensitive to UV light and have reduced levels of nucleotide excision repair. Nucleotide excision repair syndromes. They also show an increased frequency of mutagenesis, ultraviolet light, which correlates well with the patient's clinical feature of increased skin carcinogenesis. Then the zero pigmentosum syndrome has an autosomal recessive inheritance, and 7 distinct groups of genetic complementation groups were identified, named as XPA to XPG, A, B, C, D, E, F, G, 7 of them. 7 of these extinct, 7 of these distinct groups of XP syndrome are occurring. Then few patients known as XP variants, as from A to G, however, have normal nucleotide excision repair levels. May have a defect on the ability to replicate damaged DNA. So the genes related to XP have recently been cloned, except for XPE and XPV, and have revealed strong homologies with DNA repair genes from other eukaryotic organisms. So the fact that XPV and XPD proteins are subunits of the RNA polymerase II transcription factor, it is in agreement with the discovery of the preferential nucleotide excision repair of transcriptionally active genes. And this kind of repair has been denominated transcription coupled repair in order to differentiate from one pair of the rest of the genome, known as overall or general repair. So for the excision repair, it participates pre-complex after its recruitment by the protein SPA to the damage site. Again, see means that the helicase activates the helicase activity of the segment. Hello.
Hello.
Hi, sir.
Hi, sir.
Yes.
Hi,
I
sir.
think
This,
the
the
time is
screen is
almost
gone.
up. That is why the recording stopped.
The recording is still on, no, sir.
Okay. All right. When are we ending, please?
9 minutes time.
So it seems that the activities of helicase XP3, XPB, 3 to 5 direction, and vice versa, the 5 to 3 direction, may be important functions which are necessary for the role of TF2H and the homologous on the DNA repair. But XPC protein participates in the general DNA repair. The cells are mutated on the XPC gene. They carry out normal transcription coupled repair, but are defective in overall repair. So there are certain functions of that protein that it is able to bring about repair, but in totality, it doesn't repair everything. There are specific functions. So there are specific things that some of these proteins are able to do in that regard. The second human nucleotide excision repair disease is Cockayne syndrome. Cockayne syndrome, that's CS. And patients are also sun sensitive, but they show a distinctive array of congenital neurological skeletal abnormalities, including mental deficiency and dwarfism. So cells from these patients are very sensitive to UV radiation and have a defective RNA synthesis after UV. So mutations in CS, the third, please, somebody has unmuted. Kindly mute for me. It's giving me feedback here, please. The TTD, whose main aim symptoms are brittle hair and nails because of the reduced content of cysteine-rich matrix protein, and itotic skin and skull and mental retardation. Some of the TTD patients show photosensitivity correlated with a defective excision repair, but no cases of skin cancer have been related. So the TTD are caused by mutations in 3 genes, that's TTDA, B, and G. Likewise, CS, the TTD individuals with mutations in XPD may also have what XP syndromes. So the TTD is mainly a transcription disease because the deficiency of these 3 groups can be restored by the microinjection of the TF2H homologous factor through none of the known TF2H subunits is mutated in TTD-A gene. So the fact that these 3 different diseases, XPC, TTD, XP, CS, and TTD, may be mutated in the same genes as in the case of XP, B, E, and G is intriguing. So the clinical symptoms of these genetic disorders are distinct, especially concerning the increased frequency of skin tumors observed in XP, but not CS or TTD patients. These different symptoms is simply express differences in the abilities of the mutated cells to perform either DNA repair or transcription, depending on the mutation on the gene. Other human syndrome possibly linked to DNA repair defects. Additional human genetic disorders have features of genome instability that may be linked to defective DNA repair, or at least to the processing of DNA. They include Fanconi anemia, FA, and BS, Bloom syndrome, and ataxia telangiectasia. Patients with these syndromes have increased number of spontaneous chromosomal aberrations in their cells, and there is evidence for increased frequency of development of tumors early in life. So in here, you have a table depicting syndrome, main clinical features, and cellular characteristics. So FA, Fanconi's anemia, you have Fanconi's anemia, you have bone marrow deficiency, high incidence of leukemia, anatomical defects, and with the cellular characteristics, you have high frequency of spontaneous chromosomal aberrations, hypersensitivity to cross-linking agents, high recombination activity, abnormal induced apoptosis. Then with Bloom's syndrome, sunlight, hypersensitivity, butterfly lesions on the face, high incidence of malignancies, retarded growth, spontaneous chromosomal instability, slow cellular growth, DNA replication affected, Werner syndrome, premature aging, you have retarded growth, then hyper recombination, you have defective DNA initiation and chain elongation. Ataxia telangiectasia, AT, you have cerebellar ataxia, mental retardation, partial immunodeficiency, predisposition to malignancy. And with the cellular characteristics, there's hypersensitivity to ionizing radiation and some chemical agents, spontaneous chromosomal aberrations. Some of the chemical agents include laughing gas as well as colchicine. Then hereditary non-polyopsis colorectal cancer, HNPCC, you have high incidence of colon cancer in the main clinical features. Then the cellular characteristics, you have defective in DNA mismatch repair and increased microsatellite instability. With regards to syndrome, you have hereditary breast cancer, and the main clinical features, because it's hereditary, it's running through, let's say, family lines. Okay, there's high incidence of breast cancer. It may affect other organs as well. It may affect other organs as well. Then the clinical or the cellular characteristics are that cells inactivated in one of these genes are sensitive to ionizing radiation. So I think we might end up here with the lecture, and I would ensure you get the slides as well. Do you have any questions with regards to what we have done so far?
I think so, to me, I think it's okay. No,
All right.
to me, I don't have any questions for now.
Okay.
Yes, it's a bit bulky.
Yes, I don't understand why the proponents of this program decide to load it like that. Okay, it is not my doing. It's not my doing. This morning I was even complaining to some others that I don't know why distance education students have very bulky literature to battle with. And look at it, we are supposed to do 6 sessions. We are on the 3rd, and the time has elapsed. If you want to do a good job, take time to go through everything. But some others will just skip plenty things, and at the end of the day, claim they are finished. I don't handle my.
Yes, that is a challenge now, because at the end of the day, we are going out there to go and be.
So you arrive at a consensus. Arrive at a consensus, and let me hear of your concerns from Emmanuel Opoku-Agyeman and see how best I can also help you, because next week is our last lecture, and that will be on Unit 4. What about the Unit 5 and 6? How do we complete it? You are supposed to complete it. Other than that, you cannot be well prepared for the end of semester. Okay, so behind the scenes, please reach out to him and schedule maybe some weekday, maybe 1 hour, 1 hour, somewhere that I can help you out in that direction.
Okay, sir.
So on that note, I'll send the slide to Mr. Opoku-Agyeman. You make sure you give it.
Yes, I've seen it. I'll forward it right now to the platform.
All right, thank you. Enjoy your day.
Hello, colleagues. For your information, biotech has been rescheduled to 4 p.m. to 8 p.m. I think Mr. Dennis is catch up in some event that he can't excuse himself, or let's say an emergency has cropped up. So the class, he has rescheduled it from 4 to 8 p.m. So let's get ourselves prepared at that time. But for now, class has ended. I'll put the information on the platform for all to see. We were 75, we are now left with 30. You can see most have just observed. I'll put the information on the platform for us to see. Thank you very much. And then go and read your slides.
Mr. Agyeman.
Hello, Mr.
Yeah.
Hello.
Please, what of what of the Sunday schedule with? Hello.
Yes.
What of the
tomorrow
Sunday schedule
7
with?
a.m. Tomorrow 7 a.m. to 10 a.m. we are having PCK2.
Okay.
Tomorrow 7 a.m. to 10 a.m. PCK2 in biology. That is Julie, now I can now talk.
Ejopoku, please, the 4 a.m. is it the Wednesday one? The 4 p.m.
No, tomorrow. This today is today. Today's class. You know we are supposed to have class. Yes.
Yeah, after this one.
You know we are supposed to have class from 11. Listen, Julie Nyabote, Timmy. We are supposed to have class from 11:45. Is that not?
Yeah.
And he says he's not aware. He's away. He cannot meet us. So we are supposed to meet him from 4 to 8 p.m. today, not Wednesday.
Okay, I hear. Thank you so much.
Nana.
Hello, and then the lecturer was saying that we should come together with our consensus, and then we should let him hear. So as we are here, let's discuss something, so that because we cannot cover everything on the slides for this 3 or so weeks that we have. So let's come to a consensus and then present it to them.
For me, I
think what the
don't
lecturer
know.
was saying of we getting extra time for him to cover, because the question has been set already. So what we should be battling now is to get extra time for him to cover up.
There are questions, but when we were doing the mammalian anatomy, we were able to come to a consensus. Then we presented it to the lecturer. That was how he was able to break down the topics for us to have quiz 1, quiz 2, and then quiz 3. So let's come to a consensus on this one so that we present it to him as to what we will be able to learn for the quiz 1, quiz 2, and then maybe the end of semester. That is what I am saying.
Okay.
That's very good.
Julie, I think they should start questions up to unit 4 so that we don't stretch ourselves too much.
No.
We can discuss that as well now and then present it to them.
Sure.
Because this thing is so much loaded that even the way that slides are too, you have to read line on line so that you're able to get the points and then maybe get them. So if we don't channel our grievances to them for them to know that in fact it is so much loaded and so they need to do something about it, they are going to combine everything, bring them to the quiz 1, quiz 2, then we'll be there and then we'll not know what to do.
Ayeka. Monchira Marjorie.
Mr. Poku, are you around? Hello, Mr. Poku.
He's typing on the platform. I don't know why he wants to put across.
So this one there, everything is about the genetics. So if they can take units 1 and 2 for maybe our quiz 1, and then 3, 4 for the exams, and then 5, 6 for them. Or quiz 1 should be maybe something like that. I don't know.
I think I support your idea, your suggestion. Unit 1, 2 for quiz 1, 3, 4 for exams, then 5, 6 for quiz 2.
For quiz 2, so that we becomes a little bit for us.
That's all. That is it. I also support what you're saying. That's the best.
Yes.
That's the best. Because we can't combine all from unit 1 to 6 as a quiz and
exams and stuff.
It's no way. That thing too is difficult. Learning is not easy for this thing. So it's not easy. You read the car, you come back, you don't even know what you have read. Seriously.
Go around.
Technologies are too many.
Serious. Mr. Poku should take our whatever and then maybe send it to, because if not, we die finish.
He's sharing something on the page. I think the class that has been postponed to 4 p.m.
Okay, please. And then doctor said something that they said we should go and download. What was it? My network wasn't stable, so I heard some, but I didn't hear what he said. We should go and download.
It was some movie. It was a movie. So wrong time. That you said we should go and watch. That one is for your own personal thing.
Okay. Someone says a horror movie. Please don't go and watch. It's horror. That's how the one is going to turn into that horror thing.
Hey.
Is the lesson in the movie that he's asking us to?
Okay. Okay. Thank you.
Hey, I'm kind of time then. Maybe I'm you.
Did he give sites? Did he give sites we should go and download from?
Oh, I tell you when you type on YouTube,
it will come.
Holly. Please, Holly.
Wrong 10. Wrong 10.
Wrong 10.
Wrong 10. 10. TURN. Wrong 10.
Okay.
Do you imagine? I don't have time for your. Have you read your slides finished?
No, but at least. I mean, I wanted to hear what he said so that if anything, I know when they are talking about it, you know what to say. Yeah, at times, some of
I didn't hear. Thanks for raising that issue because I didn't hear.
Thank
My
you.
network was disturbing me too, so I didn't even hear.
It's network issue.
And the assignment, have you presented them? How is it in a PDF form or which form are we presenting that assignment?
Which assignment?
Next week.
Is it biotech?
The other one, the other edition.
But he said it should be in Word form. One word. And the font size should be 12.
12. Hey, so have you finished? Anthony, have you finished?
No, not yet. Still on.
In a Word form.
Yes. And the font should be 12.
Hey. I hear. Thank you.